Hello, good evening. Moshi ma'am, can you hear me? Moshi or Chitra madam, are you there? Anyone? और पार्थ सर, आप हैं यहाँ पे? मोशी मैडम, चित्रा मैडम, पार्थ सर, आप लोग कोई है यहाँ पे? Can you hear me?
Yes, sir. Yes, sir.
Okay, so I hope my, the system is working fine today, right? My audio, मेरा audio और video सब ठीक आ रहा है ना?
Yes, sir.
Okay, great. I just wanted to check before the thing actually starts. ठीक है. Okay. Thank you, Parth sir.
Yes, sir.
Okay, thank you.
Hello, hello.
Good evening, ma'am.
Good evening, sir. Good evening, sir.
Yeah. Okay. I think, yeah, some of them have, or the participants have joined actually.
Yeah,
Few
so
are
we
yet
need to look our
remaining.
resource person has joined or not.
Yeah, I just had a word with him. He's
Yes.
joining now.
Okay, okay, no issue, no issue. Okay. Yes, yes, he has. Okay.
Okay. So let's wait for, I think, 2-3 minutes.
Yes, yes. We can wait another 2-3 minutes and we can directly start.
Yes, yes, yes. I think by the time parser can transfer the presenter rights to Ratnadevi, right?
Yes, we can tell Parth bhai to transfer the presentation right to. ठीक है? So Parth bhai, आप रत्नदीप सर को presentation right दे दो.
Okay, ma'am.
जी, thank you.
Okay, Parth sir, I think Ratnadev sir is having some problem of unmuting. He has written in the chat that he's not able to.
सर हिंदी में.
हाँ, पार्थ सर थोड़ा सा एक देखिए तो आप रत्नदीप सर को आई थिंक कुछ अनम्यूटिंग का प्रॉब्लम हो रहा है. ओके, क्या आप अपने एंड से कर सकते हैं वो?
Yeah, I think it's working now. Can you hear
Okay,
me?
great, great. ठीक है,
Yes,
ठीक है.
yes,
अभी
we
आई
can hear
थिंक
you. We can hear you.
कर पा रहे हैं.
Okay.
ठीक है. I think it's 6 o'clock, so maybe we can
Yes, we
slowly
can start. We can start. Yes.
Yes. Okay, so invoking the divine blessings of Lord Swaminarayan, Brahma Sarup Sri Param Pujya Sri Sri Hari Prasad Swamiji Maharaj, Pragat Guru Param Pujya Sri Sri Prem Sarup Swamiji Maharaj, and our beloved mentor and president of Atmiya University, Param Pujya Tag Vallabh Swamiji Maharaj. I welcome you all once again to this second day of this Atal online Atal 6-day FDP program. So, as you all know, it is the tradition of Atmiya University to start any auspicious program with the chanting of Omkar. So I kindly request you all to join for the chanting of Omkar. So all of you, you can be at your place and join the Omkars. Let's begin. So, a very good evening to all of you once again. I heartily welcome everyone, including the resource persons and the participants, once again to this all AICT sponsored Atal Online 6-Day FDP program. It's our great pleasure today to have another two very eminent and renowned resource persons with us. The first is Dr. Ratnadeep Mukherjee, followed by Dr. Vijay Kumar sir. So I will know before Dr. Ratnadeep begins his lecture, I just want to give a brief overview of his achievements and credentials so far. So Dr. Ratnadeep has actually acquired his PhD in immunology from the Institute of Life Sciences, in short ILS, from Bhubaneswar, India, where he has worked on mechanisms of immunopathology in sepsis. Then he has joined Laboratory of Integrative Cancer Immunology at NCI in the USA as a postdoctoral fellow, where he extensively used mass cytometry (CyTOF) to analyze dynamic regulation of hematopoiesis and cytokine signaling networks in the mouse. He is currently employed as a scientist in the section for immunology, Norwegian Institute of Public Health in Oslo, Norway. His research mostly revolves around the high-dimensional analysis of the human immune response to infections and vaccines. His group at NIPH especially uses high-dimensional analysis platforms like multi-parameter flow cytometry, mass cytometry, that's CyTOF again, and serum protein analysis of human patients to measure post-immune response at the cellular, molecular, and biochemical levels. He then integrates this data, or his group rather, into the machine learning pipelines to derive predictive insights into the immune response heterogeneity to infections and vaccines. So with these few words about his introduction, I now like to welcome Dr. Ratnadeep to deliver his lecture. Dr. Ratnadeep, you can please share your slides and you can begin the lecture.
Yeah, thank you very much, Debashish, and thanks a lot for this invite. And I would like to thank everyone at Atme University for this invite. And apologies for the turning off of the video in between. I had to sort of run off and turn on my heater. Okay, so let's see. Do you see my slide deck or the full screen presentation?
We can actually see the, it's not in full screen mode yet.
Do you see my slides moving?
Slides are
No.
not moving, sir, but it is not in a full screen mode also.
Can you put it in the slide share mode?
That's what I did. Let's check.
Okay, it's moving though now.
But do you see the full
Uh,
screen or just like the snapshot?
No.
Sir, the thing is, which is now observing, we can see that, but if it is full screen, it is better. We can see that.
I'm checking. Can you see the full screen now?
No.
No, I don't think so, sir. It is not moving.
Okay, let's
Slides
see.
are moving, but not full screen.
How about this?
Mm,
Do you see the full screen mode?
no,
No. No. No.
no, not yet, sir.
Not yet.
Ah, okay.
Devashi, sir,
Yes.
it is observable, sir. I guess we can work out.
I mean, yeah, but then I would still like to have the full screen and let me see what is going on. I mean, I'm actually not familiar with the Cisco platform. Let's see. Share, continue.
It's true, actually. Many of the Western part of the world doesn't use this, actually.
Okay,
Even
what do
we
you
don't
see
use
now?
it. Now, I think that earlier was better because the size has.
Okay, let me see. Let me see. Do you see the full screen now?
Yes, yes.
Okay. Yeah.
Right.
Okay, these are
Yes, sir.
these
Yes.
idiosyncrasies of the different software platforms. Sorry, all right, sorry about that. So let's get started then. So what I thought I would do today is sleuthing the orchestrations of the human immune response to infections and vaccines, one cell at a time, which is basically my experiences so far, and into the exciting world of single cell immunology, and how you can sort of look into single cell heterogeneity and derive novel insights into how a human immune response or the human immune system responds to an infection or a vaccine. So I thought what I'd try to do is start off with like a brief outline and overview of single cell methods, the state-of-the-art tools that are available for analyzing the immune system at single cell resolution. And then, because I'm more interested in looking at protein expression, and that's what I do. So I'll get into a little bit more detail about single cell analysis of proteins, and then obviously a major part of the talk would be how do you analyze these data. And I mean, I think big data and machine learning and AI these are all buzzwords nowadays, and pretty much part of a biologist's toolbox. So I'll talk a little bit about that, and then towards the end, actually more towards the like halfway through, we'll see how that goes. The idea is to give you maybe one or two applications of how we are using these technologies to derive insights into biomarkers, immune biomarkers of clinical status or a particular disease. So I'm quite certain that most of you have seen this, but just like a brief overview, cells of the immune system. And let me see if I can get my pointer going. Okay, do you see the laser pointer? I'm moving it now.
Yes, absolutely.
Okay, perfect. So this kind of tree-like structure
Yes.
of hematopoiesis, as we know, which is essentially the making of the cells of the hematopoietic system, which is immune cells in blood and other tissues. Essentially, this happens in the bone marrow, where you have the hematopoietic stem cells, and this circular arrow means that they are capable of self-renewal, and then which then proliferate and differentiate into multipotent progenitors, and then you have the two different lineage, so to speak, lines of the major immune cell subsets that you see in mammalian systems. One is your T cells, B cells, and the innate lymphoid cells. The NK cells are part of that. which come from the common lymphoid progenitors, and on the other hand, you have the erythrocytes, the red blood cells and platelets, and the macrophages and the granulocytes, the dendritic cells as well, which mostly come from the common myeloid progenitors. So the lymphoid lineage and the myeloid lineage are the two different lineages, and then you have these different major lineage-derived cells. And furthermore, these cells can be differentiated into different subsets depending on what kind of proteins they express on their surface, or within the intracellular expression, for example, and depending on their functions also. Like T cells have traditionally, depending on whether they express the surface antigen called CD4 or CD8, they can be named either a helper T cell or a cytotoxic T cell. And then within these TH or TC cytotoxic T cells, you again have subdivisions depending on their functional role in a particular pathogen. If it is intracellular pathogens like a virus, then it's mostly TH1. If it is like a big parasitic nematode, for example, filariasis, then the immune response generated is TH2, and these are different subsets of cells which do different kind of things. So the idea of single cell profiling. So initially, when you look at, or let's say 15 years ago or 20 years ago, when you sort of wanted to look at the response of your immune cells to a particular, let's say, a pathogenic insult or a vaccine or some sort of in vitro simulation, then what you would probably do is take out your cell suspension and then extract total protein from it and then run a Western blot. Or if you are interested in transcriptomics, for example, then you can probably do extract RNA and then do microarray or RNA sequencing, which actually came much later, or maybe qPCR. But then it's kind of the difference between eating different kinds of fruits in a bowl or making like a smoothie of them in a mixture. Then you still get the nutrients, but then you don't really know what's inside that. So that's one of the reasons this single cell field started developing, and it's had sort of really taken off in the last 10 years. And so these are some of the things that you can actually do. As I was mentioning, microarray is essentially bulk, and on the top you have suspension-derived immune profiling, and on the bottom you have image-based, and I'll come to that. I'm actually not going to talk in detail about the imaging part of it. This is the only slide that will lead with that, but this is quite big now. So as I said, that you start with a control or a test. It can be a healthy control in terms of patient samples. It can be healthy control, or maybe a cancer, or maybe a COVID positive patient for that matter. And then you extract RNA, and then you look at specific signatures of a particular disease, like transcriptomic signatures. Similarly, then the advancement was, of course, you can do RNA sequencing then with really advancements of technologies in sequencing. This was possible, but then again, it was bulk. It's kind of says that flow cytometry was perhaps one of the only single cell tools available, but flow cytometry as such is not a new technique. It's been around since the 1970s. But it's really initially it used to be only 1 or 2 parameters, and that was about it. Maximum was maybe 4. But now, as I will mention very soon, you can actually get tens of parameters, like 10, 20, 30, 40, 50 on a single cell. And then, of course, the last 10 years, maybe 10, 12 years, has sort of seen this explosion in the single cell RNA sequencing field. And now you have, it started off with just single cell RNA sequencing, where you can actually sort these cells into different single cell profiles by their barcoding. And then you get this whole transcriptome expression profile depending on a per cell basis. And that has given us extraordinary insights that when you say 10,000 CD4 T cells, helper T cells, We initially thought of them as, before the advent of single cell methods, we thought of them as like they are like single group of cells which does something very similar. And now with this advent of single cell methods, we know that out of these 10,000 cells, each one of them are different. And that's fascinating. And every day you get to see a new paper which talks about a new subset of cells which does something else. So that's really exciting. The other thing, of course, now you can also do the same thing and take it to a slide. And obviously, like for example, a tissue section, traditionally you can do immunohistopathology, like most pathology labs in hospitals still do. Now you can take these power of these of the single cell transcriptomics and sort of have that for a tissue section. So essentially, you're thinking of like if you have, let's say, a slice of a liver, for example, like a liver biopsy, for example, maybe a tumor sample, and then you can actually look at 30,000 transcripts per cell in that, and then you basically you can sort of say that within this tissue the tumors are located somewhere, and then maybe the T cells which are attacking the tumors are on their way there, and then the macrophages are kind of helping, which are kind of in a different zone. So these kind of spatial information, and those are really exciting papers which are coming up almost, I think, on a weekly basis nowadays. So yeah. But then what I'm going to focus on today is this, and then the flow cytometry, and then of course the next advancement of it, which I'll talk about, and which we basically is kind of our bread and butter technology in the lab. So what is flow cytometry? And again, apologies if you already know this. So essentially, it's a tool for single cell profiling of any cells. Traditionally, it has been part of an immunologist's toolbox. So ideally, what you do is you have your cell suspension, could be like a little bit of blood, or single cell suspension from a tissue. Usually, it's in humans, of course, it's just blood. You cannot just go in and grab a piece of liver from someone. So and then you stain or incubate the cell suspension with antibodies against particular proteins that you're interested in. In this example, you have CD3, which is a signature marker for T cells. When I say signature marker, I mean that CD3. When you see something CD3 positive, you know that's a T cell because other cells do not express this protein, and they are present on the surface. And then again, as I said, T helper is a CD4 positive, and then the cytotoxic T lymphocytes are the CD8, so TC and TH, as I was mentioning before. And anything that is negative of CD3 are the other blood leukocytes. So then you pass them through a narrow capillary, which then goes into something called as a flow cell, where each cell passes through a single file, and it's hit by a laser. And these antibodies are tagged with fluorescent molecules, which means that they get excited at a particular wavelength of laser, and then they emit light at a different wavelength. And that light is again detected by a sensor, essentially an array of different filters. And after that, because this is an analog signal, it's converted to a digital signal, which then the computer reads. And finally, a plot can look like this, where on the y-axis you have expression for CD4, and on the x-axis you have expression of CD8. And each dot in this, we call it a two-dimensional plot or a bivariate scatter plot. And in this kind of plot, every dot is one cell. So you already see that you have right there you have single cell information, and so anything scoring high on the y-axis is obviously CD4 positive, and anything scoring high on the x-axis is CD8 positive. So you see these particular group of cells are all CD4 positive, but CD8 negative. This one is CD8 positive, CD4 negative. This little bit over here, these are the double positive cells, and these are the double negative cells. So this is kind of the benchmark that I will talk about, and that's why I wanted to show you this plot, because remember this, and I'll come back to this. This is and I'll talk a little bit about dimensionality, and what I mean by that is the number of parameters you can simultaneously measure on a given cell. In this case, it's only 2. So just these one plot of X and Y is good enough. But now I want you to think about, not think about a lot, but then just keep that in mind, that just imagine what will happen if this 2 becomes 20 or 40. How do you visualize that? And that I'll come to it. So, but there's a common issue and a problem with flow cytometry, which is called spectral spillover. Now, let me sort of elaborate on that a little bit. So this is kind of, let's say, the wavelength of like the visible spectrum is anywhere between 400 to 800 nanometers, right? Anything less than 400 nanometers, we call them ultraviolet, and anything more than 700, 750, 800 maybe, we call them infrared, the infrared range. Now, these are small molecules, fluorophores, called fluorophores. These are small molecules, and yeah, there it is, fluorophores. These are small molecules which have the property of getting excited at a particular wavelength. And then, this is high school chemistry, as their electrons go back to their resting state, they will emit that energy that they absorb from the laser, but then they will emit it at a different wavelength. Now, if that emitted wavelength falls within the visible spectrum, you can detect that as a colored light, for example. Now, all of these, so each of these different peaks that you see corresponds to a different fluorophore. Now, these dotted lines are the excitation wavelengths, which basically means that all of these fluorophores, 1, 2, 3, 4, 5, 6, about 11 of them, they all get excited optimally with at 400 nanometers. So if you shine a laser of 400 nanometers, you'll beautifully excite all these. Now, when they come back, they'll actually start emitting light, and that will be different because depending on their emission properties, and then that's got to do with the actual chemistry of these small molecules. And these rectangular boxes that I have drawn here are hypothetical detectors on the flow cytometer, which is essentially a bandpass filter. And by a bandpass filter, I mean that it's essentially like a lens like this, which can detect light passing through it given within a certain wavelength. So for example, this particular bandpass filter can sort of detect anything that is passing, that has wavelengths, let's say, for example, between 470 to, let's say, about 500 or something like that, nanometers. Anything more than that, anything less than that, this detector will not be able to detect. So that's what I mean by bandpass filters. Now, right away, you see a problem. So let's talk about this, for example. This particular bandpass filter is meant to detect this particular fluorophore. But there's this particular fluorophore also has some sort of signal falling into this detector. And this fluorophore also has some sort of signal falling into this detector. So this is basically called spectral spillover of a particular fluorophore. So it's actually spilling over into the detector into another. So for example, this guy over here, it's actually getting detected very nicely with this, but then there is a small tail of that, which is also falling on this detector. So what happens is the signal that you see, which is coming out of this detector, is actually a mix of this actual peak plus this pseudo peak, which is coming from this. Now, this is a big issue because what will happen then is you'll see something like this. Instead of this beautiful plot, which we saw earlier, like something like this, you are going to see this. So which basically means that when you try to quantify this, you say, oh, okay, so we have this 32.5% double positive cells. However, that's really wrong, because a lot of these 32.5 is just this single positive cell, which is falling into this channel, right? And screwing up your actual statistics. And same here. So what you need to do is you actually need to pull these guys back here and bring these guys back down. And that exercise is called spectral compensation, which can be done mathematically. So essentially it's like this, and this is an uncompensated data. P psi7 and APC psi7 are two different fluorophores. And then when you compensate them, this is what they look like. And you basically, what you can do is by software allows you to do that. Now there are programs written, and you can actually, even if you know how to code, for example, in programming language like R or Python, there are packages and that let you do that. So essentially, it's a mathematical subtraction of the signal of this extra signal from one channel, which is not supposed to be there. But the problem with that is it's doable in again, coming to dimensions again. I'll keep talking about high dimensions and low dimensions. This is great if you only have 2 fluorophores. Not so bad if you have 4. The moment you start going above 10, 15, 20, then you run into serious problems, because then let's say if you have 10, if you have 2 colors, you have a detector here, then you are using one color to detect so to like one fluorophore to detect one protein which is detecting here, and then another one here, and then you see obviously between this and this there is absolutely no spillover, so you don't run into these issues, so your panel design is much more straightforward. But if you are trying to look at, let's say, 15 parameters on a given cell, 15 different proteins, then you need to crowd all of that, then you need the full capabilities of the flow cytometer. So in that case, you run into these serious problems of spectral compensation. It's not impossible. People do it all the time. But it's a major headache then, because then you'll also have to keep in mind of how strong the signal is. So let's say, for example, this guy is, or rather this guy is actually falling a lot into this guy. Now, if this was what we call high abundance marker, like it is heavily expressed on a lot of different cell types, then it is going to be very difficult to take out this tail of signal. And so then what we do is we actually look at these different because these dyes have their brightness index. Not all dyes are equally bright. So the idea is, a general principle is that you look at that you sort of take a weak marker and associate that with a bright dye. And if it is a very strongly expressed marker, then you tag like a dim dye, which is okay. So these are different considerations. But the reason why I'm talking about all of that is because I want to come to this next slide then, which was basically the year 2011, and this first paper came in the journal Science from this group, Gary Nolan at Stanford University, which is essentially this particular a modification of the flow cytometry technology, which was called mass cytometry. And why is it called mass cytometry? Because instead of fluorochromes, now the antibodies are tagged with heavy metals. And again, periodic table. So the metals used in mass cytometry are essentially the lanthanide series and some metals from here. So the instrument that we have here can detect mass range from 89 atomic mass units all the way up to 209. So that is from bromine to about lead and everything in between. So what's the big advantage of this? This is again fluorescent spectral overlap, as I was talking about. Now, if you look at metal signatures, that's how they look, because it's atomic mass and we are talking about isotopes. So even if it's changed by 1 atomic mass, there is no spillover into another channels. It's like really sharp. So the grand thing is that you don't have to compensate, which is perfect. So this was a giant leap, because prior to 2011, the maximum that you could do, the maximum number of simultaneous parameters that you could measure on a given cell, was about 15, 16, and that was a very difficult panel. But CyTOF right away pushed that number to 30, 40, and now we can do even 50. Obviously, the world of flow cytometry had to catch up. So now you have spectral flow cytometry, which can do markers of like in the range of 45, 50 markers. But then those are much more difficult panels to design. And I'm not going to get into the details of the spectral flow now, because the compensation itself is a completely different ball game. It's a different method altogether. So what is mass cytometry? So it's called CyTOF by basically meaning cytometry by time of flight. So it's, the way I look at it, it's kind of a hybrid between a mass spectrometer and a flow cytometer. So this is your sample injection port. You have like a small tube here, and your samples sort of are sucked in through this, and then it goes through this line here and into this thing called a nebulizer, which kind of looks like this from a sideway. And then they go single cell profile and goes into the ICP, which is inductively coupled plasma. And then the cells are immediately ionized because of the high temperatures. So the temperature of this plasma torch is about 5,000 kelvins, and just for perspective, that's about the surface temperature of the sun. So it's really, really hot. And then, of course, there's this thing called quadruple filters, which essentially take care of your most abundant low molecular weight metals, like, for example, iron or aluminum, sodium, stuff like that. And then this ion cloud enters into this time of flight chamber, where, because they all have the same charge, they are distinguished by their size, or the atomic mass in this case, or their mass actually. And so obviously faster, so smaller atoms go faster to this detector. And then you integrate per cell and get a signature of this kind of so where like it's kind of looking at an Excel spreadsheet, where each row is one cell, and the different columns are the different proteins that you have measured. And then you can plot that into this kind of traditional flow cytometry two-dimensional plots, or we can do much more than that, and I'll come to that in a little bit. So it has wide range of applications now. It started off with only like immune cell phenotyping, looking at the different immune cell subsets that may be associated with a particular disease or a clinical presentation. But now it has sort of evolved into this different kinds of things, where you can, of course, immune responses are still the case. You can look at transcription factors. You can look at cell proliferation. Phenotype obviously is one of the earlier ones. You can look at cell cycle, inflammatory markers. You can look at metabolic markers, and this is a very hot topic nowadays, immunometabolism and high-dimensional profiling, cellular profiling of immune cells, metabolic profiling, and see if there are. So one of the things that we now know is that metabolic sort of signatures of immune cells are highly different between a bacterial versus a viral infection. So this is something that's completely novel and has come to sort of our attention over the last few years, owing to these technologies. So we have a lot of data now. I mean, I was talking about this Excel spreadsheet. In our computational terms, we call it a data frame, and maybe some of you who know how to code in R or Python are already familiar with this term. These are massive files. I mean, millions of cells, 40, 50 parameters. For site-off, it could be some thousands of cells, maybe 30,000 for if it is single-cell transcriptomics, it's fewer cells, but then the number of parameters, simultaneous parameters, go up like many, many times. So how do you analyze all of the data? And here I will sort of go away a little bit and give you a basic about some, like a very basic bird's eye overview about machine learning approaches for biomarker discovery, because that's what we are essentially doing. We are doing this high dimensional profiling to identify biomarkers of a clinical setting, like a disease or even a therapy. For example, if a drug worked, why? If it didn't work, why? If a vaccine somebody responded to a vaccine well, why? If not so well, then why not? So those are the things that we're interested in. So essentially, very basic slide. It's like data science, which is a cross-disciplinary field that seeks to extract value from data, and then you have machine learning, and a subset of that is deep learning, and of course everything falls within the umbrella of artificial intelligence. Its basic focuses on the development of computer algorithms that improve automatically through experience and using data. In simple terms, these are algorithms which enable computers to learn from data and then apply that to unseen data and sort of try to predict insights. It could be a business model, or in our case, a biological insight. This is taken from DataCamp. If you are, just as an aside, if you are wondering about how to get started in your machine learning or data science journey, this is a very good source, very good website. There are, of course, some of it is paid, but then the basics, some of the basic fundamental videos, instructional videos are kind of free to use, and it's mostly based on R or Python, but I think there's some SQL there as well. But yeah, so you project setup and then data preparation, which is obviously, this is one of the data cleaning is one of the big headaches that we all face. And then you train your model and then assess model performance. And I'll talk a little bit more about this kind of if it is sounding like Latin and Greek right now, don't worry, I'll come back to that. Hyperparameter tuning, it's one of the most important concepts of machine learning. What are the types? Of course, very broadly, you can talk about as supervised learning or unsupervised learning. So essentially, supervised learning, develop predictive model based on both input and output data. And there are kind of two different tasks, classification and regression, depending on what you're trying to predict. If let's say you have two different groups of individuals, one can be a healthy control and the other one can be COVID positive patients, for example. And your idea is to characterize 100 patients and each one of them into either healthy control or COVID positive, then that is a classification task, because your end prediction value is a categorical observable, which is in this case class label, either healthy control or COVID-19. Now, if let's say you have within these COVID-19 patients, let's say you have a disease score, which is a continuous variable, and you're trying to predict if your measured variables, your observed variables, are kind of associating with a particular clinical score, for example, then that becomes a regression task, because in this case, your prediction, what you're trying to predict, is actually is a continuous variable. And then within the domain of unsupervised cluster learning, you have this term called clustering, and this is something that we use heavily, and this is, I'll come back to that a little bit. So what is unsupervised, as I was saying? So in supervised learning, for example, you have different X's and you have Y, which is your target, and let's say each of these rows are a particular individual. And then this can be a healthy control, this can be a COVID patient, this can be a healthy control, this can be a COVID patient. So the idea here is to look at the combined profile of X1, X2, and XP. And if the algorithm is able to identify based on the combined profiles of this as to whether a given Y is a healthy control or a COVID patient. So that's supervised learning. The reason why I call it supervised will be apparent a little while later. And in unsupervised, you are not actually interested in that. You just want to see structure in the data and see if there are overall separations in, let's say, 1,000 different persons. And if there are islands, and as to how they are different, that then you can sort of come back to the supervised learning. So to start off, it's actually not a bad idea to start off with unsupervised learning methods and see if there are different like different groups of individuals which cluster together. There are many different types. I'm only mentioning a few. Regression decision tree, k-nearest neighbor, random forest, perfection machines. There's a domain of deep learning, the artificial neural networks, which I'm not going to go into today. Obviously, there have been quite a few Nobel Prizes awarded this year, both in physics and chemistry, for those particular purposes. But yeah. So what are decision trees? Essentially, they're kind of yes/no kind of questions, which can split the data into different one class or the other. For example, let's say you want to know the weather. And then a set of questions that you may ask is it sunny? Is it cloudy, or is it raining? So basically the question here is that depending on the weather, would you take the bus or walk? So you start, how is the weather? Is it sunny? Okay, so then the next question would be, how much time do I have? Do I have a lot of time? Let's say more than 30 minutes, then I would walk. But if I do not have a lot of time, then I would take a bus. Is it cloudy? Yeah, maybe. So then are you hungry? Then yes, then you can walk. If you're not hungry, then maybe take the bus and wait for the bus. And if it is raining, then there's obviously you take the bus. So these are these are decision questions that sort of let the algorithm decide as to where do I group this. And in our example, it would be, let's say, for example, you're looking at the expression patterns, which we usually do with CyTOF. You're looking at the expression pattern of a particular protein on a given cell and trying to associate that with a particular phenotype of a disease, let's say COVID positive or lung cancer. And then you see, okay, if CD3 expression is higher than 1,000, I don't know, some unit, then it can be a COVID patient. Or if not, if less than that, then it's a healthy control. So now, so you do this multiple times for as many variables that as you have measured, and then come to a final decision. So obviously, as you can see, it's very intuitive and easy to explain. I hope it was easy to explain. And then decision trees don't require a lot of data preparation as some of the other linear models require. Cons, obviously, there are obviously pros and cons, and we have to weigh in both. It's quite unstable, and a small change in training data can cause huge differences in prediction. I'll come to this, what I mean by training data. And it's prone to overfitting, which basically means that it will work very well. It may work very well with my data, but the same model will perform poorly in maybe your data. And that's one of the things that machine learning algorithms and practitioners of machine learning algorithms have to always keep in mind to really to really make their model as generalizable as possible. Generalizability is a big issue, and if your model is not generalizing, then it's not a good model, because then it basically is overfitting. So how do we get around that? One of the ways, maybe some of you have heard this term, random forests, one of the most powerful machine learning algorithms available currently. What it essentially does, as you can sort of make sense from the name itself, it's a forest. So which essentially means it runs many trees, many different trees with many different small parameter tweaking. And then what it can do is, so let's say it runs 3 trees, and for one particular sample, So this is again happening for one particular sample. Let's say we start, okay, let's say sample number 1. We train and then we train 3 different trees, and 2 trees tell me that it's class A. In this case, it could be healthy control. So 2 trees tell me that it's a healthy control. 1 tree is telling me it's a COVID positive. So then I'm going for majority voting and telling that this is a healthy control. And that's how you do it. The other, of course, the other option is you can also average. That if you are running 100 trees and you see and you take an average of the different classes and pick the class which has the highest average. So these are the 2 different ways: majority voting or averaging. Pros. And so one of the things is computationally expensive for large data sets. So you need a lot of computational power. So usually these things are done on in the cloud, or maybe you need parallel processing for this. It depends on the amount of data, obviously. So for example, for us, some of the initial stages, we need really big computers which can handle millions of rows. Like one data file could be 50, 70, 100 gigabytes. So that sort of thing. And now a couple of other things to mention in our machine learning journey. As I said, I will come back to hyperparameter tuning. So what are hyperparameters? Essentially, so this is taken from Python's scikit-learn library. And so this is the function, the random forest classifier. And the way you initiate that is basically you would just run it random forest classifier, and then within this parenthesis, if you don't write anything within the parenthesis, these are the default parameters that the model will use to run. In estimators, there's a gain in information index. Those are different criterions. Some of the things that, for example, if you would want to, let's say, how many splits do you want? How much depth that you want to go the tree to? So these are the different things, and it depends on what algorithm you choose. This is for the random forest. For something else, it would be something else. So these parameters are essentially the hyperparameters of a particular algorithm. And depending on how you choose your hyperparameters, your end result will be slightly different. So that's one of the things that you have to do is you have to tune your hyperparameters. And one of the ways you do that is by means of this method called cross-validation. Essentially, this is kind of what you do in terms of in a supervised machine learning classification or a regression model. That let's say this is all your data. What you do is you essentially split the initial data set into, so let's say this is 100 samples, 100 individuals. And you would take about 70% or 75% of those. So 75 individuals. So right away, you would remove 25 patients and run your random forest model on 75 patients. This is called training. So this is essentially telling your random forest classifier that here are some individuals, here is what their immune profile look like, and based on this particular immune profile, this can be a healthy control or a disease patient, right? Now, then you train, and this cross-validation basically means this further training data is further divided into different folds, and it can actually specify, usually is 5-fold cross-validation is good enough. So which basically means that, so for each of this split, it is going to hold out one of these folds. So for example, in this first fold, this training set data itself, it's again further training on these 4 folds. So that's 60 patients, and these 15 is again validated later on, and it's doing it multiple times to sort of final evaluate. And when you have trained your model, then what you do is now you give your model the test data, now basically saying that I'm not going to tell you what these patients are now. You look at the immune profile, and tell me what these patients are based on what you have learned so far from this. So that is really at the core of machine learning. So the machine is learning from data and then applying that knowledge to classify new data. That's basically that's kind of the idea. And one of the ways to do that is this, and I'll come back to that later. This is part from our so there are a few things that you can do to sort of evaluate your classification performance. One of the things is called a receiver operating characteristic curve, where you get a trade-off between a true positive rate and a false positive rate. And this diagonal is, and you get an ROC value, which is from 0 to 1. And this diagonal is ROC of 0.5, which basically means random classifier. Anything less than 0.5 is a terrible classifier. It's just no classification at all. And your idea of a model is to push this as much possible, as close to 1 as possible. And then you have something called as a confusion matrix. Nothing confusing about that. I'll share these slides, so don't worry about that. So you get the actual class and then the predicted class, and depending on whether there is an agreement between what the actual class is and what your algorithm has predicted from this, you can do a heat map and then project your results onto that. And I'll come back to that because I'll talk about a little bit more of that. And then you, of course, you can also calculate an F1 score. And again, you want a high F1 score. And this is essentially a ratio of true positive and false negatives. That's what TP means. TP is true positive, FP is false positive, and FN is false negative. So yes, that's a lot, but you don't have to remember everything. Just get the idea. I hope that you sort of have gotten like a bird's eye overview of how we can use single cell methods, like the basic concepts and some of the data analysis part of it. So now I'll move on to how you can apply this technologies and ask questions in human immunology. So this is my group, high dimensional immune monitoring at the section for immunology. So what we essentially do is we have a biobank where we store plasma and serum from human patients or healthy volunteers, and then you look at circulating metabolites, or in plasma and serum you can look at cytokines, chemokines, growth factors, other soluble proteins. We do a lot of this, but today I'm going to talk about mostly this. You also store the cells, and then you can look at this phenotypic state of the cells. You can look at the intracellular signaling pathways. You can look at chromatin modifications. You can also look at metabolic states. And then essentially what you do is, this is another, again, then it goes on to what we have been talking about. You do clustering, you do dimensionality reduction to visualize cells. And I'll have an example of that for cellular phenotypes. And then you also integrate the other data and do this multimodal analysis of pattern recognition, and then basically biomarker discovery, patient classification, or even disease prognosis. So that's kind of what we are doing in the lab. There are a few things that we are interested in and currently happening, exploring the immunological basis of vaccine response heterogeneity in the elderly. This is a cohort established by the Norwegian government, the Corona Vaccination Project. And we are looking at why, especially with a focus on the elderly people in excess of 65 years of age, and trying to identify why some of them have a very good response after dose 2, while some of them may need 4 doses to achieve the same response. So those are things that we are sort of exploring. We are also interested in looking at the different cellular correlates of COVID-19 breakthrough infections. So with a focus on immunosuppressed individuals, but others as well. And by breakthrough infections, essentially meaning that people who have been vaccinated have a good vaccine response, but then again came down with COVID, but actually got hospitalized because of COVID. So like seriously came down with COVID. So what went wrong? So those are things that we're looking. But what I'm going to talk about today is the immunology of post-infectious fatigue syndrome. and long COVID, and this is something that we are really excited about. We have some very nice results from this. So that's what we're going to talk about today. So immunological pathophysiology and persistent post-infectious symptoms. This is all of this work was done by my grad student, PhD student Silke. Submitted a thesis in September and then went back to the clinic. She's an MD, pediatrician, so she went back to the clinic and she was here for 3 years for her PhD in collaboration with the University Hospital in Oslo. So what is ME/CFS? Myalgic encephalomyelitis or chronic fatigue syndrome is, so now we know it as long COVID, but it has been around for quite some time. It's a disorder of persistent disabling fatigue, exercise intolerance, general malaise, cognitive complaints, and physical symptoms with significant consequences. One of the key things is that the clinical phenotype is still very poorly defined and the pathophysiology is unknown. And as a result, no disease-modifying treatments are available. In fact, most doctors sort of thought about ME/CFS as being something which is not really a disorder, but it's something that is happening in your brain. Like you're really thinking about it. It's not an actual disorder, but then this is one of the positives that has come out of the COVID infection, that we now understand that long COVID is an actual clinical presentation. It's not something that you're thinking about or dreaming about. It's an actual problem. And now people have started to appreciate the fact that post-infectious fatigue is a real thing, and we need to do something about that. So, which is a good thing. So there are few ideas that sort of that are kind of two conflicting. I shouldn't say conflicting, but it's kind of sort of opposing views of the possible mechanisms of PIFS. And this is a kind of an argument that I usually have with my clinical collaborator, which is Vegard, is a senior physician at the university. So you have predisposing factors. Of course, if you're genetically tuned to it, you may probably have it, and this is something that we all know, that it's all controlled by that. But what could be the precipitating factor is, because of an infection, you now become more susceptible to it. And the main elements of pathophysiology will be that because of the infection, you have an altered immune state, and the consequence would be chronic fatigue. There's another line of thought where it's believed that you have psychosocial factors, and then you have infections, but then the combination of that leads to functional brain alterations, and which leads to chronic fatigue, and that leads to behavioral alterations. So it's kind of a vicious cycle. And the altered immunity is actually autonomic disturbances from the brain. So kind of this A group believes that the infection caused the infection caused the immune system to activate, and then if the immune system is not able to go back to the homeostatic, the previous pre-activation normal resting state, then you can have these complications. One of that is obviously autoimmunity, and that can lead to chronic fatigue. But this other school of thought tries to think of ME/CFS or post-infectious fatigue or long COVID as a disorder of the nervous system, of the brain, where the immune functions that you see, altered immunity, is actually a byproduct of that, but not the actual cause of it. Pros and cons for both. We are still undecided, but we're kind of designing experiments as to how we can answer this question. It's a significantly difficult question to answer. It's kind of the chicken or egg problem. But there have been a few sort of papers talking about biomarkers of ME/CFS. This one was by this very famous guy, Ian Lipkin, in 2015. This is, as you can see, this is way before COVID-19. So essentially, this paper showed an increase in circulating cytokines in ME/CSF. But then what I observe here is that, so these are the different cytokines, for example, quite a few of them, IL-1 alpha, beta, interleukin-6, interleukin-12, and all sorts of different things. Now, the key point to mention, to sort of note here, is that these different cytokines have different functions in the immune system, and some do very opposite things of what the other is doing. However, if you look at this, the profile of most of these cytokines look pretty similar, and so that's why although it's a very useful study, but it's not very specific. So I think this was very early days of CyTOF. CyTOF was only maybe, so this paper came out in 2015, so this work may have been done in 2014 or something. So CyTOF was, I think, 2 years old then, so obviously not very popular. And then this was a single cell transcriptomic study much later. This is 2024. Although not long COVID, this is again just chronic fatigue syndrome, looking at monocytes and platelets and looking at their transcriptome at baseline and then symptom provocation. So by symptom provocation, I mean, I had mentioned one of the symptoms is post-exertional malaise. So basically, if you exercise, then you sort of like really, you cannot recover from that. You sort of, and it takes a very long time for you to recover from an exercise schedule. Let's say you go for a run, or in this case, it was a treadmill challenge, something like that. And these authors showed a massive difference between monocyte and platelet phenotypes and function pre and post exertional malaise. Now there's a big cohort, the RECOVER cohort, backed by the NIH in the US, doing all sorts of different phenotypic and functional analysis of post-infectious fatigue. This paper came out in 2024, last year, February, and where they did all sorts of different measure all sorts of different things like cerebrospinal fluid, autonomic nervous system, functional MRI of brain, body composition, cardiopulmonary, motor neuron activity, muscle tone, cognitive measurements, and one small part was PBMCs, which are peripheral blood mononuclear cells, which is essentially what we work with. That's the immune cellular biologist's main tool. So a lot of different things to show, but then the immune system was a little understudied, and that's one of our motivations to study this. There have been a few studies, and from what we have gathered so far is that monocytes and NK cells have been reported to exhibit alterations in proportions and surface markers in CFS-ME. Neutrophils also have been, and then there are some studies implicating B and T lymphocytes as well. In terms of long COVID, as I was saying, that after long COVID, a lot of focus has been sort of, the spotlight is on PIFS and ME/CFS now because of long COVID. And obviously, if you're an immunologist, you probably know the name of this lady. She's super famous in Yale. Her group showed in late 2023 a comparison between recovered COVID patients and long COVID patients, and a comparison with case controls, like the healthy controls, for example, doing flow cytometry, and then a lot of other plasma proteomics and antibody profiling, like high dimensional analysis. And the immune cell part was these particular subsets of immune cells were differentially abundant in the different patients. So, for example, in long COVID, they reported that this CD14 low. Debashish, quickly, how much time do I have? Is it 1 hour or 1 hour 30 minutes?
Sir, it's 7:30 you have.
Uh,
So it is
So I still
7:05
have
now.
half an hour, right? I
Yeah,
have half an hour,
you
right?
have another 25 minutes.
Okay, fantastic. Okay, thank you. So, okay, so long COVID was found to be associated with these CD14 low and CD16 high non-classical monocytes. Conventional dendritic cell type 1 frequencies decreased. So these arrows means either going up or going down. So this is enriched in long COVID as compared to the healthy controls. And then these particular subset of double negative B cells and CD4 T cells with an exhaustion phenotype are also seen to be higher in long COVID. And then there was this I think it's a consortium or something like that. This is a UK based study. Nothing cellular. This is only plasma proteomics, large scale proteomics of plasma samples. I think about 600 different proteins. And then essentially showing that inflammatory pathways related to tissue damage are implicated in subtypes of long COVID. And this is important to note because I'll come back to that. So what were the limitations? Most studies used small flow cytometry panels to address a few cell types at a time. So essentially what I've been saying, that a lot of studies looking at soluble markers in plasma with very little cellular studies and to see what kind of cellular ecosystem and how do they change in long COVID. And one of the questions that we were really interested in, how similar or different is ME/CFS with long COVID? Because we talk about, yes, long COVID is a subset of ME/CFS, but then we also need to know, so that the obvious question is, how is long COVID in a different league of itself, or is it a subset of ME/CFS, or what's happening there? So this is something that we were really interested in. So this is our clinical collaborator, Vegard, at the Akershus University Hospital. This cohort was designed long-term effects of COVID-19 in adolescents and young adults, the LOTECA cohort, which essentially has these 4 different categories. So you have acute COVID-19, and then the patients were sampled during the acute stage, and then at 6 months follow-up, where some patients recovered. So these are the COVID convalescent patients. And then obviously some of them did not recover and started showing long-term symptoms of COVID-19. And then obviously there was another group where they were confirmed PCR negative for the SARS-CoV-2, so no COVID-19. And some of them were completely symptom-free. So these are the healthy controls, and some others had CFS-like symptoms. So these are the fatigue patients without COVID. Yeah, I'm not going to go into the detail of that. So we had a lot of patients. So what we did was tackle this from 3 different angles. So the research question was, what defines the underlying immunological disturbances in post-infectious ME/CFS? We looked at circulating proteins, we looked at bulk transcriptomics, and we also looked at cellular phenotyping by CyTOF. And this is what I'm going to talk about today. So exploring the phenotype and function of circulating immune cells in ME/CFS and long COVID with mass cytometry. So these are the samples. As I was mentioning, we had these 4 different groups: healthy controls, COVID recovered, that's the RC, recovered COVID. FC is a fatigue control. So these are people who had fatigue symptoms, but were always COVID negative, never had COVID. And then the long COVID patients. And we took 20 samples from each. We seeded the PBMCs and then stimulated cells with different conditions, because we also wanted to see how the cells would behave when they're activated. And then staining of the cells with antibodies coupled with heavy metals, and then acquiring the CyTOF. Of course, you do a lot of as I was mentioning, data cleaning and data pre-processing is a huge step, and that is super, super important, not just in this, but for any machine learning or data science pipeline, because otherwise it's just garbage in, garbage out. If your data is not clean, your insights will obviously be completely faulty and erratic. And then finally, we do data analysis, which I'll talk about in some detail. So this is our panel. And as I was mentioning, 51 markers. So that's kind of what we can do now. We can actually even do more than that right now. We are designing a new panel which can do a little more than that. So these are the different mass channels. So the symbols are the different elements. For example, CD is cadmium, PR is praseodymium, ND is neodymium, and so on. PT is obviously platinum, so on. And these are the targets, which means that this antibody is against CD19, and it is coupled to 111 CD. So the 111 is the atomic mass number. So yeah, the atomic mass essentially. And this machine can detect a mass range from 89 atomic mass units all the way to 209. So it's about 135 channels in between. And right now we are only using about maybe 1/3 of that. So there's still a lot of potential to increase that. We had a lot of different intracellular markers as well, like cytokines, for example, interleukin 1 beta, TNF alpha, interleukin 6, IL-2, MIP1 beta, and so on, IL-17. So essentially, the idea is to map the global immune cellular phenotype in circulation and also look at their potential function by looking at cytokine responses. So this is the analysis pipeline. Okay. So remember when I was mentioning about the bivariate scatter plot? So when you have two proteins, it's very easy because then you can just plot them on an XY plot. If it is, let's say, 3 dimensions, you can still do X, Y, and Z, but it becomes a little difficult. But then you can have 2 different plots. So maybe XY, XZ, and YZ, something like that, 3 plots. Now, what do you do with 50? With 50, it's like practically impossible to have these bivariate plots because it will run into thousands. So what we do is something called as a dimensionality reduction technique, which is again part of an unsupervised machine learning algorithm, where what you're doing is you are taking this 40-dimensional data and projecting that into 2 dimensions. That's what you're doing. The most basic form of it is something called as a principal component analysis, which many of you may have heard. I'll come back to that like a little bit. I'll show a little bit of that. But one of the problems with PCA is that it's a nonlinear method, and which essentially means it's a kind of a straight line fitting. But a lot of these biological data it's a linear method. PCA is a linear method, beg your pardon. And a lot of these biological sciences data are nonlinear, so we need more sophisticated algorithms to really capture the differences. And this is what this is kind of the most common method of the immunologists right now is called the UMAP, Uniform Manifold Approximation and Projection. It's a nonlinear embedding dimensional reduction technique that takes these 40 dimensions and then mathematically transforms them into 2 dimensions. And I can go into the details of how that is done, but I don't think that's necessary right now. Suffice to say that what you're looking at is UMAP1. So essentially it has a set of coordinates, and that UMAP1 is some sort of combination of all of that, right? And then UMAP2 is again some sort of combination for all your markers, but then in this case, it's a slightly different combination. And then you can actually go into this UMAP1 and UMAP2 and see which markers contributed what. But that's not really super important. What we are looking at is we're looking at all our cells from all our samples again, just like the previous XY plot that I had shown a while back. Each dot in this plot is 1 cell. And what I have done here is looking at the expression of signature markers. And in our panel, it's something like this, like CD3 is a signature for T cells, CD19 is a signature for B cells, and so on. And by doing that, I have been able to then manually annotate and color this plot according to the cell type. So what you see is that this particular group of cells are the NK cells, for example, the natural killer cells. These are the B cells here. These are the myeloid cells, and this huge cluster over here, which is actually split into different clusters, are the T cells. And this kind of corresponds to what we know about human peripheral blood cells, that T cells are the majority of them. If you purify PBMCs, if it is whole blood, then it will be the neutrophils, of course, after the red blood cells. Yeah. And then this associated heat map also shows you the expression, where the expression level goes from 0 all the way up to this. So you can see the colored bar here. So for example, this particular and the different clusters are, the cluster IDs are here, and the color corresponds to this. For example, like this one you see are the B cells, and they are very low or almost non-existent in CD56, 11B, and CD3, but very highly expressing CD19, so which kind of confirms that these are the B cells. And this bar on the right shows the percentages, so 81% T cells, and which is kind of what we know about numbers of T cells in peripheral blood. What we immediately saw is that if you look at the proportions of these cells, so that's what we are targeting now. So by doing this exercise, now we want to see if the proportions of these cells are different between the different clinical categories, or in our case, the different groups, the 4 different groups that we see. And what we see is that initially the proportions of the B cells, so these are the healthy controls, and then fatigue control, recovered COVID and long COVID. So you see that the B cells in COVID, these are the COVID cases. You see that there's a slight increase of B cells, maybe there's a trend, but then it's not statistically significant, and there's not much difference in the other major lineages between the different categories. So what do? Then you dig deeper, and so if it was flow cytometry, you could only look at that, and you say that there is no difference. But because it's CyTOF, it's 40 different markers, you can actually go deeper and start seeing nice things that you would otherwise miss. So then you recluster the CD4 positive T cells, and you kind of again do the same exercise, and which now this big cloud has started splitting into these smaller subsets, and then you can actually name them by these conventional naming policies. Naive, central memory effector, and then the regulatory T cell subsets as well. And then you see that although there are some differences, maybe here and here, it's not much to write about. That you do not actually see a lot of differences between the individuals. So that's what at least the proportions of the CD4 T cell subsets. Now, if you can actually dig even deeper, because why not? And now when you see that when you further split the cells, so initially this was split into these 7 different subsets. Now you can actually, depending on the hyperparameter, remember the hyperparameter tuning, depending on one of the hyperparameters in your clustering algorithm, you can actually decide to go even deeper. And this time you're looking at 50 different subsets. And now you see that these small subsets, there are differences. And when you look at this, this is something called as a volcano plot, where essentially you're looking at, so anything that is blue, which is basically increased in healthy controls versus fatigue control, and so enriched, and anything that is red, which is decreased in frequency compared to this thing. And so if you look at it, cluster number 17 is enriched in both long COVID and fatigue. So it's probably associated with a fatigue kind of a state. And you're essentially, when you're looking at the phenotype of this, and this adjoining heat map will actually tell you, again, this is a enrichment score of the y-axis. Here are the different markers, and each cell, each block is actually telling you if that particular, so each row is one cluster. And this is just, so for example, cluster number 17 is somewhere here, where you essentially see that it's negative for almost everything that you have measured. And so I have named it as CCR6 negative CD4 effector memory T cell, because if they are negative for both CD45RA and CD197, then that's traditionally known as an effector memory. So enrichment of effector memory T cells of this particular cell subset. And again, as compared to the recovered COVID patients, the long COVID patients have an enrichment of PD1 positive CD4 effector cells. Now, this PD1 is a classic marker of exhaustion phenotype. So, for example, these T cells are essentially called exhausted T cells. So that's what this tells you. And again, a subset of regulatory T cells, which are known for their anti-inflammatory phenotype, are kind of enriched in the long COVID as well, compared to the recovered COVID patients. Now, remember I talked about also about function assessment of immune function. So so far, what we have seen is, okay, so this particular cell subset is high or low or whatever, but then how do they also behave? And that's something that we are interested in. And so we do this assessments called polyfunctionality. So which basically means that a particular T cell, if it is secreting one cytokine, so that's monofunctional. If it is secreting anything more than two cytokines, it's called a polyfunctional T cell. So T cells can secrete a lot of different cytokines. One given T cell can actually secrete multiple cytokines, and higher polyfunctionality levels of T cells is always associated with a better immune response. So we wanted to assess that. I will not go into the detail of this. If you're interested, drop me an email. We can talk about this. This is a wonderful algorithm. It can be used for other purposes also, other than just cytokines, but it was designed for polyfunctionality assessment of cytokines. So we get this polyfunctionality score, and what we see here is, compared to the healthy controls, the recovered COVID patients have higher polyfunctional response, which means that their T cells are functioning quite well, and that's probably one of the reasons why they recovered. But what we would have liked to see is that a significant difference between these two. So I would have liked to see these guys somewhere here, because then we would say that, okay, if you do not have, if your poor polyfunctional cytokine polyfunctional response is associated with your long COVID, because if you have a good immune T cell response, then you're much likely to recover. But we didn't see that. So we then actually we did a similar exercise and did look at B cells as well. Again, this subset was split into these different B cell subsets, and this is something that we observed, that the marginal zone B cells are kind of going down in frequency compared to the healthy control in both fatigue and long COVID, and a concurrent increase of double negative B cells. And this is kind of what others have reported as well. The 2023 Nature paper from the Yale group also talked about the marginal zone B cells, the double negative B cells being enriched in long COVID patients. So that was nice. I mean, that was kind of a confirmation also of our observations. We did something similar for the NK cells, and this is where we have our most striking observation. We have these 4 different NK cells, and depending on the expression of either one of these 2 markers, NK cells can be differentiated, sort of named or annotated into these 4 different subsets. And what we actually see is that long COVID patients and even fatigue patients are highly enriched in the terminal and the double negative, the CD5 sort of low, the dim NK cells, with a concurrent decrease in the early NK cells, and the fatigue control even have lesser frequencies of effector NK cells. And if you look at this plot here, this is essentially a contour plot, which tells you the density. So this is the actual cell plot. And this is the same plot, but then looking at the densities compared to the sort of comparing between the different groups. So a highly dense area essentially means that you have a lot of cells here, and sparingly dense area here means that you have a lot less cell here. So this essentially tells you that this area, which is mostly early and effector cells, right here, are highly, so the healthy controls have a lot of these. And compared to the healthy controls, the fatigue controls have a lot less of these. They, in fact, have more of these, a lot of enrichment in this area, which is essentially this terminal and the double negative area. But what is interesting to observe is that the recovered COVID patients have this in-between area where there's a lot of enrichment. So it almost feels like that because of COVID, the NK cells moved to this terminal and a terminally differentiated state, which is expected because the terminally differentiated NK cells are the most in terms of cytokine secretion, so they're required for antiviral immunity. But after the resolution of the infection, these cells should come back to homeostasis. And that's what the recovered COVID patients are showing. However, the long COVID patients are, the cells are stuck there, which essentially leads to the question, and this is something that we're really excited about, and we're thinking about how to sort of explore this further, is long COVID, is one of the consequences driving long COVID is because these cells are locked in space, locked in their place. They cannot come back to normal state. And if not, then why? Yeah, we also did this, which is they have a distinct fatigue associated phenotypes. This is again principal component analysis. So you essentially, and this is something that I was talking about earlier, that you project your n dimensions or 40 dimensions into 2 dimensions, and then you can actually look at the contributions of the individual markers in here. So for example, PC1. These are the contributions of these different markers in this score. So this basically means if you have CD161 really high, then your PC1 score will be very high. So you're much more likely to be on the right side, more on the right side of this x-axis. So that's how you read this. So for example, this one, which is really high, it is probably having a very high expression of CD161. So that's one of the ways in which you can read these dimensionally reduced plots. So we also did the polyfunctionality assay, and this is and now we see that the terminal NK cells are have more polyfunctionality, both the fatigue and the long COVID patients compared to the healthy controls. And this is important because most of these cells are positive for these inflammatory cytokines. So that kind of, now that's the working hypothesis that we have, is not only will these terminal NK cells, they're getting locked in space, but then the consequence of that is because they secrete a lot of inflammatory cytokines, they're doing so, but by doing so, they're contributing to a lot of tissue damage, which is what is probably leading to some of the features of long COVID or chronic fatigue. And this is kind of what goes back to the UK study as well, where they did mention that they did find markers of tissue damage enriched in long COVID patients. So that's something that we are following up. So in the end, we constructed a random forest classifier, as I was mentioning earlier, the test and train split. And this is something which is called as a confusion matrix, where on the one side you have a true class label, and so this is the actual class label, and on the x-axis you have the predicted class. This is something that the algorithm has learned from the data and predicted. And ideally, you should have 100%. That would tell you perfect classification. That's never the case. But this also tells you that, so for example, this, as I was mentioning, anything less than 50%. So this basically means that it's a bad classifier. So what you actually see here is that 32% of long COVID cases have been classified as long COVID, which is really bad. But then this confusion matrix also gives you an idea. I'm sorry?
Excuse me, Dr. Radhadeep.
Yeah.
I'm really sorry to interrupt you, but I think you have to just try to speed up a little bit now, because it's quite late evening here in India. and we have one
Yeah.
more speaker, so it might extend till late in the evening.
Is
Okay,
it already
thanks.
time? My clock says it's there's still
Sorry about
8
it.
minutes to go. Okay.
Yeah. So
Yeah, I'll
basically,
wind up
like,
in like 2 to 3 minutes.
1 hour,
Yeah.
15 minutes. That's
Yeah,
fine.
yeah.
That's fine.
Yeah.
Yeah. Thank you.
Thanks for a reminder. Yeah, sure. So what we see here is that a lot of the fatigue control patients are misclassified as long COVID. And then when you do a PCA, that's one of the questions. Remember when we were asking that, how is the fatigue control different? Is ME/CFS different from long COVID? And this PCA actually tells you these are the two different triangles which are practically on top of each other, kind of telling you that they're not really, probably. And so if you then rerun the random forest by combining the long COVID and the fatigue patients and just trying to define a common cellular signature of fatigue, you see the 32% classification accuracy now went up to 70%, which is like really nice. And this plot on the right side gives you a feature importance score of the fatigue, and you see that the terminal NK cells are the most important predictor of the fatigue state. So I'll actually quickly summarize decreased. We saw post-infectious fatigue associated with elevated frequencies of regulatory T cells, decreased marginal zone B cells, higher frequencies of terminal NK cells, which also have increased cytokine activated, increased activation-induced cytokine polyfunctionality. But most importantly, patients with fatigue with or without SARS-CoV-2 infection have similar circulating immune cell profiles, which is indicative of a general immune signature of fatigue that is decoupled from the initial trigger. So we are following this up, again, coming back to this brain first or the immune system first. We are kind of have some ideas in which we are exploring further. These are the people I would like to thank. This is our group here. Uni is a section director, actually the department director now, and Siri is a senior scientist associated, and I'm also kind of a deputy in charge for the mass cytometry platform here at NIH. These are the technicians and the bioinformatician. We have a bioinformatician here who is working on the data analysis pipelines. Yeah, so thank you very much for your attention, and I can take questions now.
Thank you, Dr. Ratnadeep, for such a very interesting and deeply engaging talk. Okay, on various aspects of the single cell systems, and quite a bit about the AI. Okay, the different platforms in it. Okay, a little bit of this spectral, the compensation, the spillover. Before that, okay, the little bit of the chemistry, okay, which is used in the cytop. Okay, the new applications of cytop to various areas of the therapeutics. Okay, and you also explain nicely the different types of AI, supervised, unsupervised, and there are various different algorithms. Especially your last part, trying to analyze the different factors or the different change in the immune signaling, which leads to this long COVID-like symptoms, and how in terms of that, how to use AI for differentiating different scenarios was really, really very insightful. So we take this opportunity, for all of us, in favor of all of us that are present. Let us congratulate Dr. Ratnadeep and give a good applause for this great talk.
Thank you very much. I see a lot of questions coming up, but do we have time for that?
No, so maybe we'll take a few questions now. So for the participants, the floor is now open for discussion and question answer. Though we have to keep it a little short, okay, because of the time constraints, unfortunately. So I think there are a couple of questions already on the chat. Okay.
Yeah, there's quite a few
Yeah,
actually.
maybe you can take some of them which you
I can
find
take some
more.
of them, yeah. So what I can do is, if you have questions, I'll just take some of them, maybe two. But then if you have more questions, you can send it off to Devashish, and then you can forward that, and I can answer. That's no issues at all. Okay, which fluorophore mainly used labeled with bar markers? There are plenty. Is it Ajit? Okay, yeah. There are actually quite a few, and every day. I'm saying every day, but it's an active field of research. New fluorochromes are already coming up. You also have something. It's also possible to combine two different fluorophores, which to give you something which is completely new, having different spectral properties, which are called tandem dyes. So yeah, lots of different ones. Like the first generations one, you have fluorescein isothiocyanate, then phycoerythrin with FITC-P, and then you have allophycocyanin, APC, and now you have brilliant violet dyes, brilliant UV dyes, infrared dyes. So yeah, a lot. Is it possible to identify the heart disease markers using mass spectrum? Oh yeah, absolutely, absolutely. In fact, I did not talk about the imaging site of part. I think there have been so there definitely have been lung biopsies stained with imaging site of, because you can do the same thing, look at 50 different parameters on a tissue section and get an image. So I think it has been definitely done on lung biopsies in COVID patients. I do not know about the heart, but but there are flow cytometry studies. Yes, absolutely. Is compensation ethical? Oh, yeah, it's quite ethical, but then yes, it's a very good question. And at what level? There are certain norms that you should adhere to, and also it's a good practice, and a lot of journals are currently encouraging that, and some journals have made it mandatory to share your raw files and to show how you have compensated. So yes, it's a great question, and it's very important. But yes, it is ethical. There's nothing wrong with that. You just have to sort of do it within the limits. Yeah. Okay. Yeah. MFI markers, blah blah blah. MFI markers vary with respect to cells isolated from the healthy individual belonging to different regions. Fascinating. Yes, I would seem so. In fact, there are studies. There are actually studies which looks at that, because a lot of that has to do with genetics. For example, there's a beautiful study by one of our collaborators, Dr. Maria Yazenbaksh at the Leiden University Medical Center in Netherlands. So she did a study about vaccine response in rural versus urban settings, and showed that it's completely different, the immune phenotype, depending on what you have been exposed to. Like somebody in Norway will have a very different profile, like not very different, but if you go into these details, like again, if you look at only B cell, T cell subset, like the 6-color flow, then probably you'll not see these differences. But then with 40 parameters, when you really dig deep, then you start seeing the difference. Somebody in Norway will have a different profile compared to somebody in India, for example, and that's very valid. It will be different. How can we differentiate COVID and other viruses?
Maybe I'm sorry to come in between again. Okay, between the speaker
I
and
think
participants. But since we are really running short of time, we just maybe have to take a break now. And definitely, I have already shared Dr. Rafandeep's ID. The participants can note that and post your questions directly to him. Either this ID, though this is his personal ID, but he'll be happy to take the questions. Instant, you can send the same questions to us, which will pass over to him. Okay, so I think we can, we'll stop here, and Dr. Rafandeep will definitely get back to you on, with answers on the questions. So, on behalf of Atma University, myself and also Madam, the coordinators, okay, and the scientific program, we really thank you once again, Dr. Rafandeep, for taking time out of your schedule and delivering such a very thought-provoking, lecture. I think this has really may inspire people to think about AI and how to now apply these, okay, especially in terms of disease and, the immune parameters. Thank you all again. Thank you very much again. Okay.
Yeah, thanks for having
And,
us.
Thanks. So, see you there. Yeah. So before we actually move on to the next speaker, I just have a couple of, announcement for the participants. First of all, I would be very pleased if some of you at least can put a couple of lines, about the feedback regarding today's lecture. Okay. So, this is very good for us to understand how the things have been flowing, or how do you feel about it, or is there any room for improvement. So kindly put at least a couple of feedbacks, okay, even if it is short in the chat, or you can put the same in the WhatsApp also, in the WhatsApp group also, if you like. Okay. Second, I would actually request all of you, including Ratnadeep, Dr. Ratnadeep, who is still there, to kindly switch on the video of everyone so that we can take a good group photo, which will serve as a very good memory for us. Okay, and definitely we can use that in the report also, to glorify the, to actually, feel good with all your present. So I request if it's not really a problem for the participants, can you please put on your video for just a couple of seconds.
पार्थ भाई, सब कैमरा ऑन हो जाए तो एक फोटो ले लेना ना ग्रिड व्यू में.
Participants, I
Okay.
hope, yeah, you can hear me. Can you please put on your video for a couple of seconds so that we can have a very nice, happy group photo with all of you. Yes, I think people are switching on now. I request others also to kindly switch on their videos. Yes, it's good. So,
पार्थ भाई एक फोटो ले लेना.
Okay.
yes.
Okay, sure, ma'am.
जी, थैंक यू.
Yes. Thanks to all who have already switched on the videos. Maybe, some other participants will get motivated and follow suit. Okay, I think, either they don't get very motivated to switch on the videos right now, or they may be, out of their, apart from their systems. So, I'm not reaching out to them. Anyway, we can just have a quick photo, like, with whoever video is on at the moment. So, Parth bhai, aap kindly le lijiye. Maybe we'll have a couple of other photos later. Okay.
Done, sir.
Okay. Thank you. Thank you. Some other, so participants, you can now switch off the videos if you like. Okay. Regarding a couple of other announcements, I like to say here that the 2 links for the attendance and feedback will be shared with you shortly here in the chat. I request all of you to kindly fill the attendance, especially through the link that will be shared here, because today the attendance link will be shared only in the chat and not in the WhatsApp. Please note again that you have to fill your attendance for today's session in this link that will be there in the chat itself. And we have tried to modify it so it's now clear that you'll be asked that you have attended 1 sessions, 2, or all the sessions. So you in the same form you can choose that. So I think it will be quite easier for you today. The feedback link though will be there in the group WhatsApp. You can do it till tomorrow 2 p.m. Fine. So with this thing, I hand over the platform to the respective co-coordinator of the program, Dr. Moshe Das, Madam. Madam, you can please carry on from here on.
Thank you, sir. Thank you so much for the words. Now, in the next session, I'm very delighted to introduce you and also invite our next speaker of the session, Dr. Vijay Kumar, sir, from RK University, Rajkot, Gujarat, serving as Associate Professor and Head. It's my privilege to introduce this speaker before you. Dr. Vijay Kumar have completed his MSc, PhD, and PG diploma in bioinformatics, along with qualification of ARS NET and HP SET. Beside a strong professional skill and leadership capabilities, he has various attributes which can foster various facets of his career, like being an alumni in networking with the alumni and industrial leadership, as well as building valuable connections. With strategic planning management, as well as he is also a member of different government body organizations like Probiotic Association of India, Microbiological Society of India, Association of Microbiologists India, AMI. And along with that, he has a very good number of research papers, book chapters to his credentials. He is also bestowed with the organizing various conferences, workshops, symposiums, etc. His main attribute, which we can highlight very good, that is he has delivered an online MOOC and webinar on COVID-19 awareness, contributing public health education, and his expertise in drug discovery and design, bioinformatics, and research skill are also exemplary. Apart from that, he has long been associated with us, and we are having very much privilege to have him in this particular platform to get some insights on today's topic, very enlightening topic and very interesting topic on biosensors. With these few words, I would like to invite Dr. Vijay Kumar sir to take the platform and give us few insights on the biosensor application areas. Vijay Kumar sir, the platform is all yours. पार्थ भाई, विजय कुमार सर को प्रेजेंटर राइट दे दीजिए.
Yes. Now, am I audible?
Yes, sir. You are audible, sir.
Yes, sir. Very much.
Okay. So first of all, thank you very much, Mausi ma'am, for introducing me to our wonderful audience.
Thank you, sir.
I'm in the session since the beginning.
Yes, sir.
Joined, I think 15 or 20 minutes late because I was preparing for my session. Although, I was present but not fully. So shall we start? Am I able to
Yes.
share my presentation?
Yes, yes. You are already given presentation, right? Please share your PowerPoint.
Yes. Just let me check.
Yes, please.
Is my presentation visible to all?
Not yet, sir. Please share it. Yeah, it's sharing. It's sharing. It's done, sir.
Yes,
It's done.
It's
It's done, sir.
Yes, we can
Is it in the
see
full
that.
screen mode? Is it in the full screen mode or?
No, it is in the slide mode. Yeah, now it is okay. Now it is okay.
It's perfectly fine, sir. I think
Yeah.
you can proceed.
Thank you, sir.
And I'm just changing the slides. Is it changing? Can you see the effect of changing? I'm changing the slides.
Yes. It's
Yes, sir.
changing.
Yes, sir.
Okay. So I think everything is working fine.
Yes, sir.
Yes.
Okay. That's why I wanted to check that because it happens many times that during the online sessions that the presenter is changing the slide, but the listeners on the other side are not able to see the slides changing.
It's
Okay.
absolutely fine, sir. Thank you.
Okay, so I would like to start with a small interaction with all the audience, and I don't know whether all are able to respond or not. First of all, I would like to have a question. How many of the audience present over here are using the smartwatch? Ma'am, if you can have the data, or if you can just let me know how many have raised the hands or so because I'm more of an academician and less of a researcher, although I'm doing
Sure,
my
sure,
work
sure.
in probiotic domain.
Yes.
So
Yes, sir.
messages are flashing over here. I can see many of them have raised their hands. If I'm right. Okay. Yes, yes. I can see many are replying that yes, they are wearing. They used to wear the smartwatches. So now,
Yes, sir. Yes, sir.
okay. And how many of us present over here in this session are knowing that smartwatches are a type of biosensor device? Now less numbers are flashing.
It's coming, sir. It's coming. It's flashing. Yeah.
Okay.
I think this response to the second question is a little lower compared to the first one.
I had this expectation that because we teachers, when we see in the classes and in our surroundings nowadays, the smartwatches has came into fashion since last 2-3 years. Because earlier it was prevalent to have, but now it is like essential gadget to wear these smartwatches. And I'm very much sure many of us who are present for in this session, they might also be wearing the wearable bands. They are not watches, just bands and wearable rings. How many of you are aware of wearable rings and wearable bands only? Do we have any hand raised? Okay, Dr. Saida. Sir, do we have any hand raise on this, that wearable rings and wearable bands?
That is coming, sir.
Okay,
It's
okay,
coming, sir. Many of the responses are coming, sir.
okay. Because you see, I believe more in a two-way interaction. That's why I'm initially asking the question, right?
Yes, sir. They are raising their hands. Yes, sir.
Yes. Okay. So let us start just now. That since you people are knowing that yes, there is something that is called the wearable watches, wearable rings, wearable bands, and they are not just made to have some fashion. They have some purpose. And these wearable items, wearable gadgets have came into use more after this the COVID pandemic. Because at that time, when we used to visit the malls or some places, there they used to have some oximeter that was plugged into our finger, and they used to sense that what is the temperature and what is the oxygen concentration in our blood. I hope I'm very much sure everybody of us have faced that. So that was the application of biosensor technology, which came to know public at such a fast rate, and everyone was knowing that okay, this is oximeter. Even that time, there was shortage of oximeter in the market also. If anyone has faced that, so when there was need, then people came to know that yes, this is something. But that time, these oximeters and other things which came into market, it was not came immediately. It was a lot of research was behind that, right? And this is the basis of the need of biosensor. And I really appreciate the topic which has been taken. That is the need of monitoring and diagnosing the disease. It is very much necessary to diagnose the disease at the very right time, and the course correction or taking the essential steps to correct those for the treatment of the disease. Because that oximeters and all those, they were just the indicators that the person who is using this, the person who is having the low blood oxygen concentration might be having some infection or might be having some uh, like there might be the indication that he or she may be suffering from COVID. So one who is observing these values can take the immediate step. They can take inform the doctor or likewise. So in this figure which is in front of you all, you see the person wearing the band, and the heart rate is showing there, and the blood oxygen concentration is also showing there. Even the breathing rate can also be shown on the wearables. So this is what I'm going to discuss today. That is the biosensor as a surveillance tool. And my main focus will be on the use of on the wearable biosensors, because we all are knowing that there are diagnostic kits. I hope like when we many of you once again please respond. How many of you either yourself are suffering from diabetes or someone in your home is suffering from diabetes and you used to monitor the blood glucose concentration frequently? So we have, I'm sure there will be some 50 to 60% hands raised on this. Do we have?
Yes, sir. It is. It is, and also the answers are coming in the chat box. Yes, the
Okay.
answers, the responses are coming, sir, in the chat box. So
Okay.
that we can see. Yes. Yeah.
Okay. Okay. Yes.
Yes,
So
the people can actually, answer that in the chat also.
That's right. Actually, I'm not looking into the chat box. That sorry, I don't know how to check it.
No issue, sir. We are
No.
keeping an
Yeah,
watch.
not a
Issue,
problem,
sir.
sir. We would actually relate to you the answers.
Okay. So these glucometers, they are also one of the earliest developed biosensors. No doubt, the fabrication of the instrument changes, the kits, the strips changes, but these were some of the early developed biosensors. So already there are biosensors or the diagnostic tools which are available in the market. Like there are tools for the detection of diseases. Whenever we go for the blood donation, they used to check our blood for so many, is there any pre-existing disease or not? And they all are stripped. The paper strip is there, the instrument is there. We used to put the blood on that, and after some time, within minutes, we used to get whether the reaction is positive or is negative, whether the person is suffering from some disease for which it is or not. So these are already diagnostic tools are already available in the market, but it is not available in the public, not available with the individual. It is available on the health centers, at the health centers. When we go to the hospitals or when we go to the clinics, then they used to check. And again, I would like to ask, please tell me, how many of you go for the complete health checkup twice a year? Do we have any hand raised? Do we have any hand raised? Twice a year, or at least once a year? Monitoring the complete blood profile and complete cholesterol profile, complete urine analysis once a year. Okay. Okay. Some people are raising hands. Some people are. So that is the thing that we used to go to the hospital. Then we come to know that okay, this is our profile, and then we think okay, my cholesterol level is high. We should do something. We should change our diet pattern, or we should start exercise. Or my blood glucose level is high. If it is near to the diabetic, then we go to consult the doctor. That I once in a while it came to my mind, and again I went to the clinic and I checked my blood, and I found that it is touching the diabetic level, and then the treatment starts. But the purpose of introducing this biosensors, wearable biosensor, is that you need not to go to the clinics or you need not to go to hospital. You can have at least watch on your blood profile, your body parameters, and accordingly, because nowadays many and many people are getting aware about their health. Many and many people used to go for walk. They used to do exercise. At least they give 5 to 10 minutes for the exercise. And people are shifting from junk food towards healthy food, although it is slow. But when the people suffer from some kind of disease, like if there is minor cardiac attack, then they take a lot of precaution. When they reach to the diabetic, then they used to take precaution. But it should not be like that. That is, that early diagnostic helps us in preventing the occurrence of so many diseases. And there comes the concept of wearable diagnostic, because although wearable biosensors and instruments, wearable instruments, wearable diagnostic, these biosensors, because if it is not, then hospitals are already there. So here I have some I have taken one image which is showing that what is the limitations with this the existing our existing biosensors. Like they are generally available with the clinics. They give us the diagnosis, but it is too late, or it is at least late, not in the very positive. Like there are some techniques which are now being made available for very early detection of cancer. But many of us do not go and get us checked for the cancer. So if there is something which in the blood, some wearables are there which can check our blood profile regularly, and it indicates that, oh, there is some protein which is expressed and it may lead to cancer, then immediately we will go to doctor and start our treatment. So that is the benefit of wearable devices, that early reporting is there, early identification is there, and then it helps us in controlling the disease at the very early point and get treated, and we can be cured of the disease. So again, all these pre-existing, the traditional biosensors, they are not able to identify, they are not able to detect some rare diseases. So hence again, there is a risk, and there is a high cost. Although it is nowadays more and more inventions are coming up, and it is now it has become cost effective, but still there are some rural areas where it is not approachable, where it is not people can't even afford those tests, and that's why they get late in detection of the diseases. And if you see to the looking at these all shortcomings of the existing, the conventional biosensor tools and these all kits, some smart people, they have already started doing research on the prototype development of wearable like what you have in your hand, that wearable watches, wearable glasses, wearable lenses, which can indicate the health by sensing, by checking the tears. And we have wearable clothes which can always keep a watch on our health parameter. So these all have came as an output of so much of research, and yes, there is a lot of market for that, because market potential is there is a huge market potential for the wearable devices. Everybody is now getting crazy for that. Right. So these are the advantages of this new technique, that is wearable techniques. Although I should not say it's new, it's quite old. That rapid diagnosis is possible, and it is with the user. It is not we are not dependent on the hospitals and the clinics. It is available with the user, and it can be readily integrated with the digital health platforms and mobile applications. You might have seen, you have those watch connected to your mobile, that whenever some phone call comes, you just immediately receive it. But majorly you use for that purpose. I'm very much sure not many of us have used or switched on our mobile, that why heart rate is going so high. What might be the reason? So there are the variables which are trained with this language models, this LLMs, the large language models, the AI which we know generally. So they are trained with this, that what kind of parameters are going to change, or what kind of parameters is changing, and accordingly, what might be the possibility of the health condition. So looking into that, we can immediately approach to the proper doctor. And that is what I say, that information intelligence. If we are training these softwares with the data which is already available with the healthcare people, then we can very well predict that what can be the disease, or what the steps one should take to prevent some dangerous from it. Miniaturization is another benefit. Earlier, there used to be the devices used to be bulky, but now they have come to the scale of nanometers or nanometers and micrometers. So due to this, it is easy to carry. Okay. Then we have the multiplexing capability, because multiplexing means we need not to change the variables again and again. Single variable device can monitor so many things, so many parameters of our body. So we need to invest once, and it will keep on. It will be, the durability is also there. It will keep on checking our body parameter, and by those things we develop a habit to keep a watch on our health, and we can take steps at the early as possible to avoid any kind of disease. Conventionally, this is like a biosensor. It is an analytical device that combines a biological component with a physicochemical detector. Biological components, it can be so many things. I will come next. And when the biological component interacts with the physicochemical detector or the receptor, there is the chemical reaction happens over there. That chemical reaction, it can be of different types. That chemical response is converted into measurable signals, and this signal is then displayed into a understandable information. For example, when we measure the blood glucose level by the help of this glucometer, we used to put the blood on the strip. The reaction is happening in the background, and immediately after 5 seconds or 10 seconds, we used to come to know that, okay, my blood glucose level is 90 grams per deciliter. That is, so the reaction has taken place on the strip, but on the strip number has not been generated. On the strip, some chemical reaction has taken place, some enzyme has been responsible for the conversion of something, and that release of electrons or something, that has generated a signal which has been optimized in the laboratories. This has been standardized and optimized in the laboratory, based on which some numbers have been generated, and then we come to see that if the signal has been generated to what level, accordingly our blood glucose is 90 or 95 or 100 or 110 or 120. So there must be the signal, the change of the signal must be in the understandable form. Otherwise, we laymen cannot understand this. Again, we will need the doctor to understand that, okay, this is the reading, but what does it mean? So this is also the benefit of a biosensor, that it converts the chemical reactions into understandable language, which we can understand. So as I said, like this is not very young technique. It has been the background for this has been laid way back in 1922, when we had Dr. W.S. Hughes, who developed the first pH electrode, and after that, in 1956, Clark was the person who is also known as the father of biosensor. He developed the oxygen detection method, the electrode for the oxygen detection, and this invention was the milestone in the development of the biosensor. Then we have the process goes on. There are so many things which develop later on, and then here we see this is the normal functioning of the biosensor, that we have the samples. In the samples, we can have the entire cell. It can be bacterial cell, it can be human cell. Then we have the blood sample, urine sample, or other samples we can have. So food sample, if you want to detect the presence or absence of some heavy metals or some poison or some toxins in the food, beverages, or in the air, air sample and soil sample. So these are all the samples which can be. And what is being detected? These are the biological parts. For any biosensor, there is a biological part. So this is the biological part. Then we have this is the biological part which we have. Then we have this one, the transducer, where the reaction takes place. The chemical part, the receptor for that, those are there. The nucleic acid, cells, and proteins, antibodies, enzymes are there. We can also all the biological parts, they respond, they perform some reaction over here, and those reactions are then converted into signals. That might be oxidation, reduction, generation of electrons, some variation in the electrical signaling, some generation of current, so that current is measured, or some photochemical reaction which is responsible for generation of light, or likewise So all those changes are detected over here. The electrical interface detects all the biochemical reactions. Then it is converted into the signal, and that signal, which is then converted into readable form, the signal is processed, and we use to see the response in the form of display, whether it's a number, whether it's like in case of disease kit, whether there is development of the line on the strip or not, and like this. And based on this, that what kind of chemical reactions are going on the strip, there can be, like, we can have electrochemical biosensors, we can have optical biosensors, we can have thermal biosensors. In the electrochemical biosensors, we have different types. These are the basics. I will not go deep because we are most of us faculties over here. So this is not the basic. These are not the point to be explained. Just to name this, the electrochemical biosensors, they can be amperometric, potentiometric, voltometric, conductimetric, and then we have optical, and likewise. This is the conventional one. Now coming to the present topic, which I wanted to discuss, because I thought that this topic will be of more importance if we discuss about the wearable biosensors nowadays and in the coming time. This is going to be of more use in these wearable biosensors. So you see here in this image, you see the biosensors, the wearable biosensors. They can, since as the word is saying, the wearable, we can wear it. It can be a device or it can be our clothes. It can be our shoes. It can be our socks. Okay, these are all. It can be a chip. We can just place a chip on our skin and it will keep on monitoring the thing. And how it works basically, the wearable biodevices, they are in constant touch with our skin. Let me just show you. It is not placed on the right place here. You see here in the this is the anatomy of the skin. Here on the uppermost layer we have the stratum corneum, the epidermis, because we have epidermis, we have the dermis layer, and this one is the layer which is in touch with the wearable device. Now, the wearable devices, if they are based on the micro needles, I will come on this later on this topic because micro needles I need to discuss in detail. But if they are the fabric, the cloth which we have, or we have the patches which we can just put on our skin and it will keep on. So all these, the fabric, all the patches, or the micro needles, they are in constant touch with the epidermal layer of the skin. And below the epidermal layer, we have this interstitial fluid is present in the viable epidermal. And in case of microneedles, they are patched within. They just get embedded within the stratum, and from there, as you can see here, the dermal layer is there. The fluid, the body fluid is here, and the body fluid is the best indicator of anything, any minor change in the body. Whatever change is taking place, the body fluid get that change very early, and that's why now the scientists are working on it to the non-invasive methods of detecting the diseases, because nowadays we have so many things have been developed, non-invasive techniques, because earlier the invasive techniques were there, people used to be fear of that. But nowadays we see in among the students that if we want to ask them to check your blood group, many students used to fear, no, I have the fear with fear of needles, or I have the fear of blood, I can't see blood, I can't bear the pain of punching and all. So that's why the non-invasive techniques are more important. They do not cause any pain. They are just in touch with our skin, and whenever there is change, they detect it, they process it, and they send the data to our devices or to some to our health professionals. We can have the consultation with them, and they can have the course correction on time. So, okay. So here is that is a simple comparison of this. That although the conventional biosensors, they offer the valuable insight, but they are available with the laboratory. And POC diagnostic, that is the point of care diagnostic. These point of care diagnostics now presently it used to be the clinics or the health centers. but it is not needed that they can only be the point of care center. Nowadays, our wearable devices are being converted into point of care devices, which is collecting the information from us, and they are processing further. So this is the main, as I said, non-invasive method. This is the major advantage of the wearable biosensors, because conventional biosensors, there is pricking, but in case of this, there is no pricking. Okay. So, and the flexibility and comfort, that is also when you are wearing any device, the patches which it is with you, it will not annoy you. You can keep on, you can wear those patches for long durations, and it is not going to make you any discomfort. And these wearable devices are user friendly, they are skin friendly, they are very soft, because they are like for textiles, the wearable textiles, they are very made of a soft fabric, they are breathable, they will not irritate you, and they will not cause you any allergy. And since it is to be weared, they are flexible, whether it is embedded in your socks, whether it is embedded in your clothes, it is flexible. That's why they are easy to carry on. And major benefit is also that you can by the help of wearable devices, we can maintain a continuous watch of all the parameters, because it is in continuous touch with the skin. This is the limitation, that in case of conventional systems which is available, it cannot monitor it continuously. We have to we can monitor by regular intervals, like we used to monitor our glucose level, but it is at regular intervals. But nowadays, the patches are available in the market, which is you just need to patch it on your shoulder, and by changing the color of that, you can get the information whether your glycemic level is changing, whether it is increasing after it, like even after a meal which you take, and just after 10 or 15 minutes, you can see by changing the color that which diet is responsible for the change of your glycemic index. You might have seen the ads of those devices nowadays. Okay. So wearable devices, they can be of different forms. As you can see here, this is I have just taken this image from somewhere in the net, and I have also provided the materials which I'm using here to ma'am. She will be sharing with you all. Okay. So starting from our shoes to our head, every wearable device is now available that can help you monitor your health at any given point of time. The shoes, they are embedded with wearable biosensors which can monitor the sweat released by the shoe, the pressure which you are putting, how you are walking, and likewise. So because they have the motion sensors, the rings in the fingers, as I told earlier, the watch which many of you are already wearing that. Then we have the strips and the bands, which always keep monitoring. They will not, that might not be connected with your mobile, but yes, and even they can be connected with your mobile, but not for the purpose of picking the call or so, because for that we have the watch. But these are going to be going to monitor you regularly for your heartbeat. It can tell you whether you are going, whether you are feeling hypertensive or something. Then what is your breathing rate? What is your pulse rate is going on? Then we have the clothing embedded with the biosensors that can also monitor so many parameters. Then these mouth guards which monitor the saliva and any abnormality, any presence or abnormal presence of any component, biochemical thing in the saliva, that can also be get monitored. Then we have the glasses, the contact lenses are also there nowadays. They can also watch monitor the tears and the secretions, and accordingly they can indicate. Then we have the headset. So these are all the wearable devices which have been developed. These are all have been developed. This is not that will be developed. These are all in market. Many people are using these. Okay. This one is an extension of what we are just discussing, that how these are all being discussed, like how are these are all being watched. Okay, like we have you might have seen in this image, you see the mask is there, and this mask is not a simple mask. They are also having the and they have the ammonia sensors, and there are so many such things which are there which monitors uh, the gases which are coming in the breath, and according to that they can sense that what can be the consequences or what is changing in the body. Then this is already the blood sensors which are already available, but now they have been converted into the patches. Then we have the pulse monitoring devices which we use to wear, and then we have there are so many real-time monitoring devices which as in this image you can see, which helps us in monitoring our entire parameter at any given time. Okay. Now, okay, before coming to this, although I haven't included, ma'am, I have a PDF. Can I share that? With the help of which it will be easy for me to explain the thing.
Like, sir, the PDF, like materials you are asking?
Yes, because some images are there in the material which I would like to share.
Which particular material you are asking? Just let me know. I will try to make it happen. Like the PDFs which you have shared in that, which particular material you are, or
Okay.
all the.
No. Can I? I have changed my screen. Is it visible?
Yeah. You can also share from your side. Just
okay.
You have to say share any other. Just you have to say share any other. So you can also share. In the share screen, you can see share any other. Like that you can see. And apart,
is it visible to all? I have changed now in front of you, whether it is my presentation or something, because I have opened the PPT, opened the PDF material over here. Is it visible to all?
Sir, just a second. I just confirm. PDF share hua hai?
No, sir. It's not actually shared till now.
Okay.
We can see the PDF only. Yeah.
Let me just see if I can. How can I do? Because when I was going through, so some of the content which I have not added in the PPT, but they were important, which I just wanted to.
You can do, sir. It is possible. Just click that option, share any other or share other, like that in the top red box it will show.
Okay.
Otherwise, sir, like you can try that. What ma'am is suggesting, if it doesn't work, the other option I think is to just stop sharing this one, and you can share that other one. Okay, maybe you can switch again.
Yeah, that is also
the window has been minimized, and I'm unable to maximize it.
You do one thing. You just stop share. You can see 2 plus equal sign at the top. Just stop share.
Okay.
Do stop share and again reshare what you wanted to share. Just stop share.
Okay. I find it. I found it.
Yes. Yeah.
Okay, I have changed. Has it changed there?
Yes, sir.
Yes, sir. Now
Yes, sir.
show it.
Yes, sir. Yes, sir. It is there. Wearable sensors images in the video. Yes.
Yeah. Okay. Yes, this is what I wanted to have. Because there's so many information in this material. Out of like I have gone through some 7 or 8 materials, but out of those I found only 3 materials which are more important from today's point of view. So I have, and it was not possible to bring everything in the PPT. So that's why I just thought that I should share this. So the image which I was showing in this PPT, it was taken from here. And you see the wearable devices, like I want to give an insight onto how it is made and what materials are used for the fabrication, because the fabric is an important point, because for different purposes, different fabrication materials are used. Now you see, like I will just keep this in front of you, and accordingly we can have Okay, there is one. Is it changing, ma'am? I am changing the materials over here. Is it changing or it is fixed? Because 3 PDFs I have kept open. So is it changing there or not?
No, sir. Yes, yes. Now it is. Yes, sir. It is.
Like,
it is, sir. It is.
Micro needles are here. In front of my screen. It is micro
Yes,
needles.
micro needles. Micro needle fluorescent sensor. Yes, sir.
Yeah. Okay. Thank you. That is what I wanted to confirm, that if I'm changing over here, is it changing there also or not? So I would like to show one image that was there. Sorry, I'm taking time, but please, in order to show this, it is, yeah.
It's visible, sir. It's absolutely visible.
So you see, I wanted to have this one, the D image. Okay. So how this fabric, the fabric which we are talking about, the wearable devices, the fabrics are made. So the selection of fabric material is also very important, right? So it has an active layer for any fabric, any wearable fabric. It has an active layer which is in touch with the skin. Then we have this in between layer which performs all these chemical reactions, and then we have the outer layer which is which processes all the information and senses. So the active layer which responds to the various stimuli because it is in touch with the skin. It can be made up of metallic nanomaterials. Like the major role comes here is of nanoparticles. The gold nanoparticles and silver nanoparticles, since they have a very good conductivity. So metal nanoparticles like gold and silver, they have very good electrical conductivity, and thus due to these electromagnetic properties, they are of very high importance and they are very frequently used for making the nanomaterial outer layer. Then we have the other material which is used, that is the carbon nanomaterials, like carbon nanotubes, like graphene. If you are aware of that, graphene, they are nowadays graphenes have come into discussion among the public domain in like common in last 2-3 years. Graphene. They have they also have very good electrical conductivity, and their chemical stability is also very good, and bio compatibility, due to which it is since it is made of carbon, and that's why they do not show any allergic responses, or majority of us will not feel any difficulty due to that. And by modifying these nanotubes, carbon nanotubes, it can be given any shape, and thus they are of much importance when it is when the matter comes to integrating it with the fabric. Okay, so that's why the carbon nanomaterials, carbon nanotubes are of high importance. They are preferred materials for the use. Ma'am, if you will be providing all these materials to our audiences, they will be much benefited with this.
Sure, sir. Sure. It will be shared to them. Okay.
The third material which is very much preferred, that is the conductive polymers. The conductive polymers are also frequently used for the purpose of preparing this fabric. And this is about the outer, the active layer. We have this beneath the active layer. We have the interface layer, which is responsible for connecting and transmitting the signals which are provided by the reactions, the biocompatibility layers of the outer layer which we have. Whatever, because it is in touch with the skin, so whatever interactions is being there, that is transferred to the interface layer where this interaction takes place. And now this interface layer, that is made up of the polymeric materials like biocoagulable materials and the bioactive nanoparticles. These are they can be charged, they can be provided with, they can be tagged, they can be added with the chemicals and the enzymes where the reactions can take place, and that's why they are more or less preferred. Since I don't have that, I have tried my best to explain using this, but what I'm saying, that much material is not over here. But even then, I hope you people are getting, because Can I have just whether the people are getting what I'm saying or not? Because when we used to have the habit of seeing the people and knowing their faces, whether they are getting or not. Ma'am, can I have that?
Sir, it is already in the chat box, few responses. So I guess they are at par with your informations, and they may raise questions after your lectures, obviously. So
Okay.
there are questions. Can
Okay. Now returning to my presentation from this point, we will come back again to this point. Yes, it is. So why I went to this fabrication part, because just to give you a sense that there might be questioning raising into that into your mind that we are saying that the biosensors, but how they work. Now there's a deep physics and chemistry in that. I'm also not that much aware of that, but yes, at this point of time, to understand something about this was the thing which I wanted to have. At least you should have that the interface is made up of conductive materials, and beneath that layer we have the like in case of strips. No, in case of the strips we have the biological material, then we have the transducer, then we have the signal modulation, and then we see. So here also similar things are there. Ma'am, is it in the full screen mode? Yes.
it is in slideshow actually.
Okay. Is it?
Yes, it is now full screen.
Okay. Now, coming to the application part of the wearable biosensors, because we just saw the different parts. Now,
Sir, can you just minimize that particular magnification, like let it be in full screen? Yeah, now it's okay. Yeah, it's okay. In the mobile mode, somebody are there. So now it's okay, sir. No problem. It's okay.
Okay. It is in full screen mode now. Okay. Now, before coming to this, the wearable devices have been made with the carbon nanotubes and all. These things have made it very easy and accessible, and as compared to the expensive devices, it has been made very easy to access, and anyone can, as per their need, they can afford these techniques. That is the main importance of the development, the innovations that they are made accessible to all. And these textile-based biosensors, they can definitely they can embed it into the textile, so it can analyze the tissue, the pressures, due to our different postures, due to our body motion. They can analyze the pressure, how much pressure at which point of the body is being made, and accordingly they can give you the real-time analysis. And this is mainly important for sports person. They used to wear also. And these textile-based biosensors, they have the piezoresistive sensors that offers very simple preparation and integration with the devices. And the signal processing is also very it can be readable and it can be understandable form. Then we have a sweat sensor, again, since it is in touch with the skin, the sweat, which is now, wait a minute, yes, the sweat glands, which are just in the dermis layer, they are very important. And again, the sweat glands, the secretions of the sweat gland, they can also give a lot of information about our body, about our body condition. So the sweat glands, the sweat being secreted over there, they are being tested and they are being checked over there. Let me just go to that content. Yes. sorry. Along with this, after this, we have the temperature and humidity analysis that can be done with this wearable biosensor, and thus we can have this the idea of so many different parameters of this. Now, there is another kind of wearable biosensors, but they are not that much comfortable as the wearable one. I have one comparison of this. I will just first go to this comparison that the wearable biosensors and the microneedle based biosensors. The microneedle based biosensors are both are wearable, but compared to the textile biosensors, the microneedle biosensors are sometimes they are because the microneedle based biosensors they are not non-invasive. They like the patches, which I was just talking about the patches. The skin patches, they have the microneedles which embed into the skin. The textile-based biosensors, they do not get embedded into the skin, but they remain they remain in touch with our body always. But in case of the microneedle-based biosensors, they get embedded into this. And yes, for that I want to show you this image. You see this. The microneedle-based biosensors, these are this. This is the structure of the microneedle. Here is the skin, the outer epidermis. So they are punched in, and they are not going deep into this. They are not going deep into the body because their length is hardly between the 250 to 1000 nanometers. Okay, they are a kind of wearable, but they are the needles. It doesn't mean that you will feel the pain. It is painless because they are so small that you may not bother whether it is stick on your body. So they are in the range of 250 to 1000 nanometers, and they penetrate the skin's outer layer. And from this outer layer, they have the access to the interstitial fluid. This one, the fluid present in between the epidermal layer here, because the nerve endings which are opening over here, they also secrete the chemicals, the glands which are there, and the fluid which is present in around the cells. They are all available over here, and due to this, they can the microneedles have this access to the interstitial fluid, and from that interstitial fluid, they can check the concentration of different salts. They can check the concentration of hormones. They can check the concentration of other body fluids, and accordingly, they can give the information. Right? Like these are the some analytes which can be of the priority, like it can check change in the pH, it can check change in the concentration of sodium, potassium, and fluoride, and so many things. Lactate formation, is there any formation of uric acid, and other things, they can be checked with the help of microneedle. And accordingly, these are the related disease conditions which can be indicated by the help of these patches. Okay. So these are depending upon the condition of the disease, there are different types of patches are available which can be selected for wearing or which can be selected for being stick on this skin. Now, this is one representation which I have taken from one article that how these reactions take place and how this works. So in this image you can see this one is the microneedle. It is again coated on the hydrogel, which will be forming the base of holding these needles and which will stick to the skin. Then they have in this layer you see they have this red donor, red acceptor, and the receptors over here. These are the analytes. There's a small yellow and red and purple color. These are the analytes. When these analytes bind to the receptors, they produce the signals which are then analyte receptor complex. They are formed and they produce the signals which are then detected, and it is sent. The message is sent to the connected devices on which we get the information. These are the structures of this. The microneedles, how they are formed and how they appear in the when we see it. So these are the, as you can see over here, the fabrication of the nanoneedles and the microneedles, which are of this. They you can see all these. These are the very small ones, so they do not penetrate much deep into the skin, and that's why they they do not give us any sense of pain. Inside these needles, we have the entire electronics over here, on which the reactions the, uh, the reactions take place, and we get the information on it. This is some wearable device on which we can, as you can see here, these are so tiny that here is the image of the skin after putting it. So it is in the microscope. It is not going to make any damage, much damage to the skin, and that's why they are nowadays preferred also. Coming to the continuous application of these microneedles, we have a simple application, drug delivery, and monitoring of various biomarkers, and of course, glucose monitoring is very common for any kind of biosensors, because diabetes is a world disease. A lot of cases are coming up. Many. It has been so common. And here's a comparison of this: the textile-based needles and textile-based biosensors and the microneedle-based biosensors. So, ma'am, I have compiled my presentation. I know I have went very fast, but this is all from my side.
No, sir, it's absolutely fine. And you have tried to compile in a nutshell, in a very lucid manner, starting from basic to taking to the advanced and the research areas very beautifully. So, participants, we can take one or two questions.
Yes, yes. How can I see the questions?
Sir, you can see in the chat box. And participants, if you have any question, you can raise your hand. We will unmute you. पार्टिसिपेंट्स अगर आप में से किसी को भी क्वेश्चन पूछना है, आप अपना रेज हैंड कीजिए. ऑप्शंस दिखेंगे आपको सिस्को वेबएक्स में, जो लोग मोबाइल से यूज कर रहे हैं. सर, अजीत कुमार सर हैज आस्क्ड अ क्वेश्चन.
Yes, it just flashed, but it went.
I tell you, which biosensors can we choose depending on accuracy for glucose measure?
Okay.
The continuation is, I just complete it, because the same person using glucometers from different companies will get different readings.
Different readings, yes, yes, it is. Okay, so in this, the glucometers are just giving the approximation, because for exact measurement, whenever you go to the doctors, they perform the biochemical test. But yes, for continuous monitoring, the microneedle-based biosensors are more preferred for the glucose monitoring compared to the it is also wearable. So nowadays, doctors, for those who are diabetic, now the doctors are suggesting that whether to go, you should go for some, like they comes in the patches form. They are microneedle based, and that's why they keep the continuous monitoring of the glucose level. Okay, so yes, microneedle based biosensors are more preferred for that.
Sir, the next question from Sakina Rakhiv. Any study on wearable devices to assess the stress, like for example cortisol level?
Yes, yes, it is. There are wearable devices for the stress level monitoring, and it is already in the market.
The another question, sir, from Ajit Kumar again. Is there any standard for selecting biosensor? Which company product give high accuracy?
No, not exactly, but like there are so many companies, like G is also making the device, and that is all based on the reliability, and there are data available in the research papers and all, which compares these devices from time to time, and those which are more near to the laboratory results, they can be selected. But yes, based on the past studies and the comparison studies performed by the researchers, then only we can choose. Otherwise, going to the market and bringing anyone, that will not give the exact there used to be that 5 to 10%, 5% error is always there. That's why those brands which are much established in the market and they are proven for their quality check and all, they can be selected.
Sir,
Sir,
I hope there is no questions.
yes.
No, I
Yes.
think someone is
Yeah.
there. Yes,
Sir,
sir. Please, yes,
actually,
sir.
I just have a question to you. So we are looking at this biosensors and speed based tests, but some people actually don't rely on them because according to them there is a certain degree of variability. According to your opinion, how much you feel is the variability between a lab-based or the clinical biochemical test and these sensors, and if at all these there is a considerable degree of variability between these, what might be the factors behind these, or in a sense how reliable for a suppose for a management of a patient who's critical, can we rely on these kind of biosensors and based kits or this other POC based kits?
Sir, biosensors are diagnostic tools. They will give you the diagnosis of what is changing. But for the treatment purpose, it is always recommended that biochemical tests must be performed in the laboratory under the supervision with matching those data with some standards. Like as you said that if you are in the process of designing a device for, say, for example, of monitoring the blood glucose level, so you have to match those results with the standards, and then only you have to bring in more and more accuracy of your device so that it can give as much result near to the biochemical test as much possible.
Right. So, I asked because this till now, maybe in the Western side, all these latest technologies, they are more, adopted or rather faster there in the Western side. In Indian context or Asian context, I think if the condition of the patient is critical or emergent, they are in an emergency, people actually, rely much more on still on the lab-based test or biochemical test.
Yes,
Okay,
and everyone
these kind of
in the rural areas there is no much availability of those laboratories. So for those conditions, as I said, that many research and many people also have it has been established that if we have more and more availability of these biosensor devices for diagnosis, for monitoring the disease condition, then based on that, those patients can be brought to the hospitals or clinics earlier.
Right. Sir, one last question is that,
Yes.
Sir,
got
can
your
we
point.
take the participant questions before?
Yeah, please.
Yeah,
I'm-
sorry
I'm
for the
sorry,
interruption, sir. Dr. Isha
Sure.
has asked Vijay Kumar sir,
Yes, ma'am.
what are your views on PPG marker?
Sorry, Isha ma'am, I am not right now. I am not aware of this, but yes, definitely I will find it and I will provide you the response.
Sir, maybe I'm bothering you, but sorry, another one question from Rohini Tikale.
Ma'am, please mail me or provide me the answer or a question of Isha ma'am. I will find it and I will provide you the-
Sure, sir, no problem. I have already taken that. Sir,
Thank you.
Rohini Tikale has asked that is there specific any biosensor which are used to monitor blood glucose level in children?
In children.
Yeah,
Okay.
it is Rohini Tikale is asking.
In case of children, there are there's a fluctuation in their glucose level. So, patches can be used, patch-based, the microneedle-based biosensors can be used to monitor. But yes, since there is high fluctuation in them, so it cannot be you need to know that what is the condition. Whether if the children are suffering from diabetes because it can be genetic, then accordingly, yes, we can monitor the condition, and patch-based biosensors are more preferred because multiple time pricking in case of children that will be painful. But microneedle patches based biosensors, they will be. I think doctor will prefer that only.
Thank you, sir, for the answers, and I hope the participant has got their answers. And Devashi, sir, sorry for the interruption. You can just continue with the questions which you were asking.
No, it's fine. It's because this topic was equally very interesting, and
Yeah,
that's
because
why
sir, so many questions are coming. I couldn't just keep myself or like prohibited enough. So
yeah.
If you have any question, please let me or. But yes, please pass me the questions which I am unable to answer.
I have already done in your mail.
Okay.
So
Okay.
so it is the time, I guess. Participants, you have, if any more questions, I am going to share with you the mail ID. So not to worry, because for two interesting topics, first one for Dr. Ratnadeep sir, and second one for Dr. Vijay Kumar sir. It is really the heat of the day, and I guess in this chilly night, still people listening and patiently and just on the online platform. I guess the interesting topics are taking the brainstorming things in their mind, and definitely they will think today and tomorrow because it is a Sunday. So definitely they will think the morning time for this. So with this particular things, Parth Bhai, we can have a round of applause for congratulating our resource person Vijay Kumar sir.
Ma'am, a question just flashed, I think, which was about the multi-channel.
Sure, sir. I will mail you,
Would
sir.
like to answer that these multi-channel microneedles, they have
Yes.
the variability, and thus they can be used for different diseases at the same time. That was the question was linked to that, I think.
Yes. So any things we will definitely share with you in your WhatsApp and mail ID. Parth bhai, one round of applause, please. ताली हो जाए थोड़ा सा. Thank you so much, sir, for this insightful talk. Devashi sir, would you like to conclude the session?
Sure, ma'am.
Thank you, sir. Thank you for the.
So thank you, ma'am, and thank you, Dr. Vijay Kumar sir again for his very interesting talk again on the biosensors and all. So all this kind of things, every we are looking at is throwing some kind of very, some interesting and most of the time some unknown information. Okay,
True.
though we have all heard about biosensors and all, but we really didn't know the, application part of it, or how these are so much related to our daily life. So, we really are very grateful to you, sir, for again taking time out of your schedule and, agreeing to our request to deliver your lecture here. We definitely, as discussed here, we would pass on your contact information to the participants, and they would be able to relate with you if they have any more doubts or for any kind of collaboration with you in future. Once again, from the entire Atmiya, the FDP team and the Atmiya University, we thank you very much. Okay, and we hope to, collaborate with you again soon on these kind of platforms.
I'm also thankful to your team and the Atal scheme for giving me the opportunity to share my little knowledge on the biosensors and all. And thank you,
It's not
everyone.
little, sir. It's quite big, and it really has triggered our minds for it.
Devashi sir, could you find a voice? I guess somebody are asking to raise questions. I just thought maybe can you see once? Maybe somebody is asking from back, like asking question.
If we would request the participants actually to either you can if you have still any questions, you can still post it in the chat, or you can raise your hand so that we know it clearly that you still have some doubts. So kindly raise your hands if you have any other questions.
Okay, I guess it's just no more questions now.
Right.
So we can just yes, sir. Please, your concluding remarks.
So we with this we come to the end of the day 2 of this ATL sponsored 6-day online faculty development program. Today's sessions were equally very interesting, and once again we are deeply grateful to both the speakers who have delivered such nice informative talks. So we depart today at this point, and again tomorrow we hope to see you all again for a nice interesting day again. Thank you all. Thank you the participants also for having the patience to keep joining, okay, till now. So that shows your interest. That shows really your interest, okay, and we hope the same kind of interaction and motivation again in the forthcoming days. So once again, good night to all of you from here, and see you all tomorrow. Goodbye.
Thank you, sir. Thank you, Devashish sir, for coordinating this. Thank you, participants. It's a very delighted experience to have you. The good numbers. Thank you. Thank you, sir. Thank you so much. Good night, all of you. पार्थ भाई बंद कर दीजिए